Well, good morning, everyone. And this is Rick Wycheck, the director of NAEHS, and I'm joined by members of our council and the senior leadership committee. I want to welcome all of you to our council meeting. So I'll start off by saying that pursuant to the Government in the Sunshine Act, all aspects of this meeting are open to the public except the review, discussion, and the evaluation of grant applications and related information. So David, I'm going to turn it over to you. You're going to take us through and do some introductions?
I will indeed. Thank you, Rick. Good morning, everyone. Welcome to the NAEHS Advisory Council meeting for February of 2024. So I'm actually going to begin with a little bit of Zoom etiquette, just to remind folks that are on the Zoom. This is a fully virtual meeting, so we have no in-person component today. And people have been doing Zoom for the last few years, but just a reminder that this meeting is being webcast. Staff and members of council are on the Zoom. I would like to ask everyone to keep themselves muted and also to keep their cameras off just for bandwidth's sake. We're going to be using the raise hand feature for moderating question and answers. So you can use the raise hand, please, to be called for a question and answer and discussion. Keep your VPNs turned off, again, for bandwidth issues. There will be times when council members need to be absent from the discussions for conflict of interest reasons. In those cases, we will put you into a waiting room in the Zoom, so you do not need to leave the Zoom. We'll just exit you into the waiting room and bring you back when we move on to the next topic of conversation. And members of the public who would like to express views regarding any items discussed during the open sessions may do so by sending an email to council using the address listed on the council webpage, which is national.advisory.council@naehs.nih.gov. And also a reminder to council members that we will be conducting votes, and those will be conducted electronically using the electronic council book. With that, I will start with introductions of our council members. The only council member that we know of that is not going to be present for council this month is Phil Bourne, who is on international travel. And I'm just going to go through alphabetically and call on council members to introduce themselves. So our first is Dr. Greenemeyer.
Hi, I'm Tim Greenemeyer from the University of Pittsburgh. I'm a neurologist and neuroscientist interested in gene-environment interactions in neurodegenerative disease.
Great, thank you, Tim. Dr. Hertz-Picciotto.
Hi, everybody. I'm Irva Hertz-Picciotto. I'm at the University of California at Davis. I'm a professor in the Department of Public Health Sciences, and I direct the Environmental Health Sciences Center. My research is lately primarily on climate change and health.
Dr. Holian.
Hi, I'm Lee Holian, University of Montana, Director of Center for Environmental Health Sciences. My research area is in respiratory toxicology and mechanisms of chronic inflammation.
Dr. Hood. Daryl, you're muted.
Sorry, that happens on Monday. I'm a professor and Dean's Fellow here at The Ohio State University. I am an environmental public health neuroscientist, neurotoxicologist, and I'm the co-architect of the Public Health Exposome. So you can guess what my favorite area might be. Thank you very much.
Thank you, Daryl. Dr. Hornbuckle.
Good morning. I'm Carrie Hornbuckle. I'm a professor of civil and environmental engineering at the University of Iowa and director of the Iowa Superfund Research Program.
Dr. Ingram.
Good morning. This is Janie Ingram from Northern Arizona University. I'm a chemist And my background or my research is in environmental health studies on tribal lands.
Dr. Leveist. Do we have you? You may not be on yet. Dr. Miller.
I'm Gary Miller. I'm a professor of environmental health sciences at Columbia University. I work in exposomics and I'm a neurotoxicologist.
Dr. Mutlu.
I am Gokhan Mutlu from University of Chicago. I'm a professor and chief of the section of pulmonary critical care. I'm a pulmonary critical care physician. My research focuses on how particulate matter air pollution causes lung inflammation and cardiovascular effects.
Dr. Nez Henderson.
Good morning, everyone. My name is Patricia Nez Henderson. Greetings from Rapid City. I work for the Black Hills Center for American Indian Health and been focusing on addressing issues when it comes to commercial tobacco and ceremonial tobacco. Thank you.
Dr. Penning.
Hi, I'm Trevor Penning. I'm from the University of Pennsylvania. I direct the Center of Excellence in Environmental Toxicology, which is a P30 Environmental Health Sciences core center. My area of interest is in hormone and chemical carcinogenesis, with an emphasis on using analytical chemistry.
Ms. Sylvester Kennedy.
Hey there. Sorry. I'm Maria Savasta Kennedy. I'm a clinical professor of law at UNC School of Law. I'm the lawyer in the group here. And my area of teaching and research is environmental justice. Good to see you all.
Thank you, Maria. Dr. Vasquez. Karen, I saw you on earlier. Yeah.
Hello. No, I'm sorry. Hello.
yeah.
Yes. Sorry about that. I was having to sign off that I'm okay that we're being recorded. I don't know. Anyway, good morning, everyone. Karen Vasquez. I am from the University of Texas at Austin, and I'm professor and division head of pharmacology toxicology and leader of molecular oncology in the Livestrong Cancer Institutes. And my research interest is on understanding mechanisms of DNA damage, repair, and genetic instability in cancer and other genetic diseases. And sorry about the pause there.
No problem, Karen. Thank you. We have 3 new members who are joining council. They're in the process of finishing up their paperwork, and we received permission to have them join council as non-voting ad hoc members. So I would like for them to introduce themselves. The first is Dr. Cormier.
Good morning, everyone. I'm Stefania Cormier. Most people call me Steph. I'm with Louisiana State University. I am the Associate Vice President for Research and Economic Development. I am a Wiener Endowed Chair and Professor at LSU, and I'm Director of the Louisiana State University Superfund Research Program. And I look at early life exposures and how they lead to respiratory health effects later in life. Nice to meet you all.
Welcome to council, Steph.
Thank you.
Dr. Hoyo.
Hi, my name is Catherine Hoyo. I am at NC State in the Department of Biological Sciences. I'm an epidemiologist, and my research area is on environmental epigenomics and metabolic outcomes.
And then the third is Oliver Deshane, who is going to be joining us this afternoon. He was not available this morning. And then we have our ex officio members. These are members of council representing other federal agencies in providing advice to NAEHS. They are non-voting members. The first is Yulia Carroll.
Hi, I'm Yulia Carroll, and I am with the Centers for Disease Control and Prevention. I'm the Associate Director for Science at the Division of Environmental Health Science and Practice. Our focus is on applying innovative data to detect environmental public health hazards, building the environmental public health evidence base, and also developing and implementing best health practices. Thank you.
Thank you, Yulia. Andrew Geller.
Hi, I'm Andrew Geller of the Environmental Protection Agency. Office of Research and Development, where I'm a senior science advisor. My background is in experimental psychology and toxicology. These days, I'm focusing on environmental justice and lead toxicity. Thanks, David.
Thanks, Andy. And Dr. Fitzpatrick.
Oh, yes, this is Dr. Susie Fitzpatrick. I turned my camera on, but I got a terrible cold. So I am a senior toxicology regulatory scientist, and I chair the FDA's alternative methods work group. So I work mostly on new approach methods for FDA.
Thanks, Susie. And now I will move on to a review of confidentiality and conflict of interest procedures. So policies and procedures for preventing conflict of interest and confidentiality of grants materials are contained in the conflict of interest forms, which have been emailed to members of council this morning by Liz McNair. That document states that this certifies that in the review of applications or projects conducted by the National Advisory Environmental Health Sciences Council on February 12th and 13th, 2024, I absented myself and I did not participate in the discussion of nor vote on any applications or projects in which I, or to my knowledge, my spouse, minor child, or close professional associates have a financial interest, nor on any applications or projects from an organization or institution where I am an employee, consultant, officer, director, or trustee, am negotiating for employment, or otherwise have a financial interest. In council actions in which my vote on a block of applications or projects without discussing any individual one, the en bloc actions, my vote did not apply to any applications from any institution fulfilling the criteria in the preceding paragraph. Members should sign the certification and email them to Liz McNair, actually, we're not going to leave them on the table since we're all virtual. Please email them to Liz. And we will move on to our first action, which is the consideration of the minutes for our September 2023 meeting. The minutes are available on the NAEHS homepage, and council members were asked to review the minutes prior to this meeting. They are also included in the electronic council book. May I please have a motion from council to approve the minutes?
So moved.
Thank you, Maria. And may I have a second?
I'll second.
Thank you, Gary. And if council members could please vote. Do you have quorum, Liz?
Not yet. Still waiting.
So sorry, can you remind us where we would find the voting for this? I'm in the council book, but having an issue.
Beside the entry that says the category that says council minutes, it says view, vote on documents. A hyperlink that says right beside it says vote on document, click there.
Thank you.
We have quorum.
Thank you, Liz. And now I will turn it over to our director of our Division of Intramural Research to introduce our first speaker. Daryl.
Thank you and good morning. So it's a pleasure today to introduce Dr. Shanshan Zhao. Shanshan is a senior investigator in the Biostatistics and Computational Biology branch here at NAEHS. She received her PhD in biostatistics from the University of Washington in 2012. and completed her postdoctoral training at the Fred Hutchinson Cancer Research Center before joining NIEHS as a tenure track investigator in 2015, and she was promoted to senior investigator with tenure in 2022. So the overarching goal of Dr. Zhao's research program is to develop powerful statistical methods to discover how human interactions with the environment influence health and well-being. She and her research group employ a dynamic approach by integrating statistical methodological developments and collaborative population-based studies to reveal the social and physical environmental determinants of human health. They develop specialized statistical methods for disease risk assessment and prediction with multivariate time to event outcomes, which are now the standard methods for analyzing this type of data. Motivated by collaborations with other NIEHS researchers, she's also developed novel statistical tools to address issues that frequently arise in cancer and environmental epidemiologic studies. So today she's going to highlight a series of new statistical tools that she developed to characterize the spatial and temporal distribution of cancer incidence and mortality in large cohort studies. Without further ado, let me turn things over to Shanshan.
Thank you, Daryl, for the introduction. Can everyone see my slides? All right. So the title of my talk today is called Empowering Environmental Research Through Novel Statistical Methods. So I don't think I need to convince anyone that environment has a large impact on human health. And I want to start with a couple figures on the WHO's website that they summarized the pathway how environment impacts human health through water, through air, water, chemical, radiation, noise, and a couple work and life-related exposures, as well as climate change. They also estimated about 24% of all global deaths are linked to the environment. Of course, the impact of the environment is different at a different part of the world, as well as for different gender and age groups. So to understand the impact of environment on human health, a lot of important population-based data have been collected, and I want to highlight two studies here. The first is the sister study sponsored by NIEHS, and this study collected over 50,000 women who has at least one sister with breast cancer, but were cancer-free themselves at baseline. And up to last year, this study has already followed the participants for 20 years. Another program that has an even longer history is the Surveillance, Epidemiology, and End Results, or SEER program at NCI. They celebrated their 50th anniversary last year. And this program continuously collects and analyze data from population-based registries. And they cover about 48% of the US population. So with these amazing efforts being put to collect population-based data, as a statistician, I view it as our responsibility to develop accurate and powerful statistical tools to give insights about these data, so to test for hypotheses, and then also to generate hypotheses to guide future research. So today I want to show you two projects that in which we contributed to environmental research through developing important statistical methods. So in the first half, I'll talk about how we worked dynamically with our epidemiology collaborators to provide a set of tools in assessing disease risk in both space and time, and then to relate these observed patterns with environmental exposures. And in the second half, I'll give you a very brief overview of some statistical methods we developed for environmental mixture analysis. So my first project is motivated by the sister study, which I already mentioned. It is a prospective cohort study with over 50,000 US women with at least one sister with breast cancer, but they never had breast cancer themselves at baseline. And these women were enrolled between 2003 and 2009. So the primary outcome of the study is time to breast cancer in the scale of age. And up to 2020, we observed 3,820 breast cancer cases. This study also collected a lot of important risk factors, including individual level demographics and reproductive and medical history of these women. They also recorded the enrollment residential address, which allowed us to look into the impact of social and the physical environment. So I was first involved in the study through a collaboration in this White et al. Paper, where they looked at the impact of air pollution on breast cancer in the sister study. So in this paper, they used a well-calibrated kriging model with spatial smoothing to estimate the individual level PM2.5, PM10, and nitrogen dioxide exposure for each woman. And then they used a Cox proportional hazard model adjusting for baseline demographic information and these exposures. So they found that PM2.5 is associated with elevated risk of DCIS, and nitrogen dioxide is associated with elevated risk for both overall breast cancer and the DCIS. They further went on to look at the spatial differences of these relationships. So they categorized the women into 4 regions: Midwest, Northeast, West, and the South. They also looked into the PM2.5 component, because that's PM2.5 is a summation of 22 components. So they used the method previously described in Keller et al. 2017 paper to define 7 clusters of the components. And here, these colored dots are where these clusters are located at. And you can see there is a very obvious spatial difference in terms of the composition of PM2.5. So their analysis, when clustered by region, found that both invasive breast cancer risk and the DCIS risk association with PM2.5 vary across different regions of the country, and the spatial heterogeneity is significant. And also, that is also the same situation when they stratify by component cluster. Of course, the sample size in some of the clusters is a little bit too small to give you a reasonable confidence interval. But my take-home message from collaborating in this project is that there is important spatial differences for breast cancer risk, and some of these differences can be explained by the environmental exposures in different regions of the country. So after that, I asked myself one question. So can we directly model the disease map of breast cancer incidence first, and then relate it to the environmental risk factor as a different approach to decide what important risk factors we want to look at first, and then look at the spatial difference. So I presented this question to my postdoc at that time, Dr. Rachel Carroll, who has a lot of experience doing disease mapping. But her answer to me is actually quite surprising. She told me most studies are using aggregated count data for disease mapping. That means if we want to use the existing method, we need to aggregate our individual level data into counts, which will be sacrificing a lot of information. We also searched in the literature. We found a few papers talking about using individual level data to model disease map, but most of them are using binary outcome, which means they take a snapshot in the time and look at whether a woman has developed breast cancer or not at that time point to do the disease mapping. We think that also sacrifices a lot of important information we have using this prospective cohort. So this type of outcome we have is called time to event data. Some of you might have encountered those data in your study before. Basically, this is a diagram showing what's complicated outcome situation we will have. So for everybody in our study, because this is a prospective cohort, so we won't get to observe their experience before they start the study. However, if they developed breast cancer before the start of the study, then they are not eligible. And then we get to observe their experience during the study. So for those 3,800 women who develop breast cancer, we know exactly when their breast cancer event happened. But for most of other women in the study, we only know that their breast cancer is either in the future or won't even happen during their lifetime. So that's called right censoring. So was the special feature of time to event outcome was left truncation and right censoring. Some special statistical inference procedures are needed to make inference. So this is the model we decided to use. It's called accelerated failure time model. So I already told you about a Cox proportional hazard model that we used for the previous study. And the Cox model, some of you might know, is most common method people use for time to event data analysis. The reason we decided to go with accelerated failure time model instead of Cox model is that the Cox model has some embedded assumptions about proportional hazards which will prevent us from further adding in the spatial and the temporal distribution. So anyways, the accelerated failure time model, the form is here. It's nothing more fancy than a linear model because we just model the log of the survival time t, which is in the scale of age here in the study as a linear function of covariance x. But because of the special features of t here, you need to know that there is a there are some special statistical tools that are needed to make inference, but we don't have to open that black box today. We just need to know this is the basic model. And then in terms of if we want to add the spatial component to model the disease map, we propose to add in a spatial random effect W here, and we can impose special statistical structures for this spatial random effect so that we allow women in neighboring or within the same spatial area are more alike than women in a faraway area. And we did all the inference under the Bayesian framework and all the regular model building procedures such as model selection and confidence intervals. Those are valid under this framework. And all the details can be found in our 2017 paper. When we apply this to the sister study cohort, this is the spatial variation we observe after we adjust for individual level risk factors. So here, each unit is called a public health region. This public health region is usually defined by each state to combine a couple counties into one region for logistic or administrative reasons. And the reason we decide to go with this spatial resolution is that we need sufficient statistical power. And once we do this, our sister study participants are represented in all 382 house regions in the contiguous US, with an average of 117 participants and 10 breast cancer cases in each region. So how do we read this map? Here, the darker color corresponds to a smaller spatial random effect, which indicates younger age at diagnosis or increased risk. Basically, these darker areas are at a higher risk for breast cancer. And from this map, we notice very significant spatial patterning. Mostly we see lower risk in the south and the southeast, and a greater risk in the northwest and certain areas of Midwest and the northeast. So in order to interpret the environmental exposure, in order to interpret the spatial distribution we observe on the map, our collaborator have considered a list of environmental exposure that may help us to interpret those spatial differences. So these exposures are based either on previous study or literature. So for example, the PM2.5, NO2, and PM2.5 composition, which showed important contribution from the previous study, are also in this list. They additionally added UV radiation, green space, total and anthropogenic ambient noise, outdoor light at night, and population density, as well as area deprivation index. These exposures all have very well calibrated models, and they are calculated at individual level for each individual woman in the sister study. So these variables are all some of them are highly correlated. For example, these 4 here are potentially all related to urbanicity. So we couldn't adjust them all in one model. Then we decided to go with the forward variable selection to pick the most important variables. So our best model has additionally included area deprivation index, nitrogen dioxide, and light at night. And we found ADI and NO2 are significantly associated with the outcome, while light at night is marginally significant. So with these 3 variables added to the AFT model on the left, that's the model without adjusting for those 3 exposures. And on the B panel, that's the one adjusting for environmental exposures. We can notice that the pattern we observed before is much less obvious, with most of the part has a good mix of these colors. There is a little bit of remaining spatial variation at the Midwest. And quantitatively adding those 3 exposures explain away about 21% of the spatial variation we observe in the A panel. We also did a similar analysis, but only restricting to ER-positive breast cancer cases. And this is the panel C is the map without adjusting for environmental exposures. And the clustering effect is very obvious. After we adjust for environmental exposures, this pattern is almost gone. And those environmental exposures explain away about 63% of the total spatial variation, which is a lot. And all the details can be found in our 2023 paper on environmental research. And this paper is also selected as NAEHS Papers of the Year 2023. So basically for the sister study data, once we finish with the disease mapping analysis, our collaborator has a list of important environmental exposures for us to consider to interpret the observed spatial heterogeneity. So we kind of did a hypothesis testing type of analysis. But what if we have little knowledge about the relationship between environmental exposure and a different health outcome or in a different cohort? Then think about a genetic study where you could test a lot of genes at the beginning. Then we may also have a long list of potential exposures from publicly available data sets such as EPA air pollution data. And there's a lot of summary of the environment from, say, National Center of Health Statistics. So we could look at a long list of potential exposures, and this list can even grow in the future. So we thought about statistical learning methods such as machine learning or deep learning method to accommodate the high dimensionality and high correlation. But because of the model structure being put up to now, we haven't been able to do that yet because of the computational burden. But that might be a future research area we will explore in the future. But anyways, we started with a simple idea. We want to perform a test that is relatively simple, such as a t-test for each of the exposure for discovery purpose. So this is what we plan to do. For this illustration, I'll switch to the SEER data, which I've already mentioned that this SEER program collects cancer incidence and survival data from population-based cancer registries. So for each case, each cancer case, they collect very detailed individual level demographic and clinical variables, as well as their county code. And they follow these cases up for their vital status, which allows us to model time to death. So for this study, we specifically chose the SEER Louisiana breast cancer mortality data from 2000 to 2013. This data set covers 48,000 breast cancer patients from all 64 parishes in Louisiana. We chose this data set because Louisiana is very diverse in social and physical environment. And a couple notable events happened during this time period, such as Hurricane Katrina in 2005 and the passage of Affordable Care Act in 2010. And we are also interested in understanding how these events affect the breast cancer mortality. So when applied the proposed spatial model of the AFT model, this is the map we have for the Louisiana data. And this is how we read the data. We identified Santamini County with Santamini Parish, where New Orleans metropolitan area sits in, as the longest lived county, while Terrebonne Parish is the one with the shortest survival. So for a woman in Santamini Parish versus another woman in Terrebonne Parish, if they match on all the individual level risk factors, the woman in Santamini has 1.66 times longer survival time than a woman in the Terrebonne Parish. So this is a very big difference. And so how do we understand this big difference? So we propose to categorize these spatial regions based on these estimated spatial random effects by quartiles. Basically, Santamini Parish and some of these lighter colored parishes will belong to the longest lived category, while Terrebonne and a couple other very dark areas will belong to the shortest survival category, with two more categories in the middle. So for each of these potential environmental exposure on our long list, we can test whether there is a significant difference between these regions in different categories. So we call this as a secondary assessment of the environmental exposure effect. And what we perform is very similar to a t-test, but remember these parishes are, some of them share border, so they are not independent of each other. So we accommodate correlation between these neighboring areas, and all the details can be found in our 2018 paper. So here is a partial list of the important risk factor in environmental factors that we looked into, comparing the longest versus the shortest quartiles. And here is a partial list of results that I focused only on the social related environments. And we found that these two categories differ a lot in terms of access to healthcare, quality of healthcare and their socioeconomic status, the average socioeconomic status. So this is very interesting result to us. But now we want to switch gear to understand a little bit more about time. Why do we care about time? Because there's general trend related to knowledge, education, disease screening, and other reasons. And some of the time windows have notable events happening, such as natural disasters or health policy changes. For example, this is a figure I got from the SEER website, looking at the mortality and the incidence rate of breast cancer from 1975 to 2020. So for mortality, we notice a slightly increase in the early 1990, and then a gentle decreasing trend over time. But for the incidence risk, we notice a increasing trend from the 1975 all the way to about 2000, followed by about 5-year decrease over time. And then there is a slightly increasing trend from about after 2005. So we think a lot of these trends are related to the general knowledge, education, treatment, better treatment options. But this decreasing trend is very interesting. There is a hypothesis that's related to the publication of Women's Health Initiative's hormone therapy trial results that greatly reduced the prescription of hormone therapy in women. So I don't want to talk too much about the trend, but I just want to use this as an example to say time is really important. And then for some diseases, the time trend can even interact with space. For example, for COVID incidence rate, definitely the time trend is very different for different part of the world. So we came back to this accelerated failure time model where we already added in a spatial random effect. Now we further add in a temporal random effect, but we have two here. The first one is the one that representing the general trend. So that's related to continuous education or improvement of treatment. And then we added one more that is called event-related temporal random effect, such as the one related to the WHI hormone therapy trial publication or natural disaster. So our model can even accommodate more complicated spatial temporal random effect to accommodate the interaction. And again, we did all the inference under the Bayesian framework, and we used the deviance information criteria to select the best fitting model. And for the event-related temporal random effect, we have two versions. One that we can supply to the algorithm, say which event we are interested in looking, such as Hurricane Katrina or Affordable Care Act, or we can let the data to explore which event is the most significant and what is the time window this event is in effect. So this is the result for the Louisiana data. On the y-axis, that's the temporal variation, basically larger temporal random effect correspond to longer survival time. So you can see the general trend is that the survival rate is increasing over time. But for the dotted line, this represented the most significant effect in the time window when it's in effect. So we found that the most significant event impacting the survival time for women during this period is definitely Hurricane Katrina, which happened here in 2005. And the effect is lagged by about 14 months. And it lasted for about 2 years and 7 months. This is a very interesting result to us. And I think this dip corresponds to potentially women at that around after the Hurricane Katrina hits, has a lot of things that they need to take care of. So it delayed their delayed their treatment or diagnosis. So that's why we start to see a drop in the survival rate or the increase in the mortality rate about 14 months after that. Just to summarize, here we worked dynamically with our collaborator to propose a series of statistical tools for us to characterize the spatial and the temporal pattern of disease risk. And we further related those patterns with environmental exposures, both descriptively through the map and quantitatively through secondary assessment. And these methods, although motivated by breast cancer research, can actually be applied to other types of diseases. Now, in the second half, let me give you a very brief overview of some statistical methods we proposed for environmental mixture analysis. So we are very interested in the mixture analysis because we believe all humans are exposed to an array of chemicals, and previous studies have been mostly doing single exposure analysis, but the results could be very misleading because these chemicals usually interact synergistically or agnostically with each other, and they are never acting alone. And we view the mixture analysis as a very solid step to move from a single exposure analysis to exposome analysis. And on the right side, we summarized these are the scientific questions that mixture analysis usually asks. And NAEHS has this great funding program that called Powering Research Through Innovative Methods for Mixtures in Epidemiology, or PRIME program, and to fund research related to the mixtures. And then here is a summary of some specific questions to be addressed in mixture analysis, including identifying the most important toxic agent, estimating the overall effect, identifying patterns of exposure, as well as accommodating interaction and nonlinear relationship. We also want to incorporate some predefined scientific grouping into the analysis. So with these different aims, a lot of statistical methods have been developed for each of them. Some are specifically for mixture analysis. Some of these methods are more general for omics type of data, but have been accommodated into environmental mixture analysis. And a lot of these statistical approaches are trying to address the high dimensionality and high correlation challenges that are common to many type of omics data. But we started with a slightly different perspective. We go to the fundamental assumptions about some of these studies, some of these methods, that they all assume very accurate measure of these chemicals. However, in reality, these chemicals are never measured very accurately. They are always subjected to some technical difficulties, technical limitations. So here we specifically looked at chemical measures subjected to limit of detection. For example, in this Life Codes cohort, which is a prospective cohort of women in the Boston area who plan to deliver at the Brigham and Women's Hospital between 2006 and 2008, we are interested in understanding the relationship between 17 urinary trace metals and 8-isoprostane, which is a very important oxidative stress marker for pregnancy. However, among those 17 urinary trace metals, only 3 of them are measured on everybody. All other 14 chemicals are measured with certain percentage of values below LOD, ranging from 0.8% to about 91%, which could lead to very different results when we handle them differently. So in the literature, people have been using several popular approaches, including complete case analysis, substituting the values below LOD by certain values, such as LOD divided by square root of 2, and multiple imputation, which is more computationally difficult. And we consider most of these methods do not consider the correlation between the exposures directly into the calibration approach. And they don't really take into account the uncertainty in calibration into the final outcome model. So what we did was because the left truncation is somewhat similar to the left truncation issue we see with time to event outcome, and we have developed standard approaches to handle multiple time to event outcome before, and we apply that multivariate AFT model to innovatively to this type of multiple exposures subject to limit of detection in the first stage. And in the second stage, we jointly model the outcome and exposure to accommodate the variability in the first stage for calibration. And we conducted both case study and an extensive simulation study on how these different LOD correction methods can be affecting downstream mixture analysis. So I don't have enough time to talk about the details, but just to summarize that our finding is that quantile-based mixture approaches are less sensitive to limit of detection corrections, but they can be very biased if the quantiles are not maintained. And our proposed two-stage AFT model performed very stably and accurately, followed by multiple imputation. And all the results and the details can be found in the Lee et al. Paper that is currently under review. So we have also conducted, we are currently working on several other projects in a mixture setting. For example, we have a multiple index interaction model that considers scientific grouping of chemicals and calculate one overall index for each group, and then allow interactions through these indices, which offered ease of interpretation. We are also working to bring the Bayesian additive regression tree, which is a very powerful tree-based method in the literature, to the field of mixture analysis because of its computational efficiency. And we are modifying the algorithm so it can accommodate nonlinear relationships, scientific grouping, and a variable selection into the method. With that, it concludes my talk, and I want to thank all my NAEHS collaborators, Dr. Lexi White and Kelly Ferguson, who I worked very closely for the two projects, and Dr. Dale Sandler and Dr. Claire Weinberger are the PIs of the sister study, always offer very great suggestions and support throughout my project. And I want to thank all my current and former trainees, Dr. Rachel Carroll, Lingwan Chen, Matt Lee, and Kai Zong Yi, who did all the detailed work for these two projects. And I'm happy to take any question.
Thank you, Shanshan. We have time for a couple questions. See, I see a couple of hands up. Irva.
Hi, thank you. Very interesting presentation, Shanshan. Really appreciate that work. I just had a couple of questions about the analyses you did on with these various methods for spatial and temporal variability assessment. And one of them is I'm thinking about the prior period before the start of the study, and whether you had any kind of data about earlier life exposures or locations, or any changes in, it was any data collected on changes in locations going forward, because it seems like a lot of what you're looking at in the time course is sort of short-term effects on breast cancer as opposed to the longer term effects that there's been a lot of emphasis on early life risk factors for breast cancer and other cancers.
Yeah, I think that's a great question. So far, we have only looked at the enrollment residential address, so we can estimate the average exposure a year before the enrollment. But I believe we do have childhood residential address or the longest lived address available, and that's something we are interested in looking at. But to be honest, I think the childhood residential address may not be the best approach, because it can change a lot. I think we collected the childhood longest lived address, but not really covering the whole profile of where these women are. I think maybe some recalling of whether they lived in a farm, have exposures to the pesticides and other things can be added into the analysis to simultaneously address the issue of environmental exposure. But I think that's something I need to talk to my epidemiology collaborators to see what their inputs are. But that's definitely a limitation of our current data analysis.
And would you be able to do more than one
For the
location,
future?
two different spatial factors?
I think that's yeah, I think that is a possibility to not even not just to focus on one time point, but kind of looking at the spectrum of the time and see where they have been, and kind of do a more detailed kind of calibration model to take into account the whole history of the residential address. That's definitely something interesting. I think the difficulty would be first is the accuracy of data. Second would be statistically, how do we incorporate all those information, especially how long they lived in one location, into account. That's a little bit unknown to me right now. I need to look into that.
Thank you. Thank you.
Thank you.
So we have time for one more question. I don't see any hands. So let me just ask a question about generalizability. So the spatial and temporal models that you've developed, have they in fact been applied to other diseases and or other cancers other than breast cancer?
Right. Yeah. We actually looked into the colorectal cancer using the SEER data again. And we're interested in that because we were at one of the talking about little is known for the trends. So we looked into the SEER Iowa colorectal cancer and looking at both mortality and incidence to see how they differ from, especially in terms of time trends, and also for different age groups above 50 and below 50, because I think the screening is different for that. And we did notice some different changes across time, especially when the screening guideline for men are changing from, I think, 55 to 50. Then there is a big pickup of the incidence for younger onset of colorectal cancer. So those are also published in one of the paper on Clinical Colorectal Cancer Journal. So yes.
Hey, Gerald Hood.
Thank you very much, Shin Chen. Very nice talk. Thank you. Along the lines that Irva was interrogating you on, was just sort of wondering, I was very happy to see those results for cluster 4 after having worked with the Southern Cohort Community Study for a decade or so. So it's nice to see that concordance there for sure. But now what about, and I think Daryl just sort of touched on this, I was interested in what this might glean for some of the other gynecological cancers. You mentioned the SEER data and NCI they were very, very high on residential history, for an example. But are you able to extrapolate a little bit, if you would care to do so, regarding some of these other gynecological cancers?
We haven't yet. So we have been mostly focusing on the developing the model within the breast cancer kind of context and apply that to the colorectal cancer. I really, if you have any idea of what other type of cancer may have this kind of dramatic changes over time and at different locations, I would be very happy to look into that. As statistician, we really rely on the experts at different subject area to let us know where there needs to be some special accommodation of statistics there. So yeah, I welcome any suggestions of where we should look.
Thank you very much. Great talk.
And I see a question, last question from Andrew Geller. Andrew, do you want to unmute yourself and ask, or do you want me to read it?
Sure. I thought we were ending time, but just quickly, I see in your analysis of the Louisiana data, you looked at percent agriculture, forestry, fishing, hunting, mining industries. Does that also include the chemical industry? We know that the chemical industry is very active in Louisiana, and there are a lot of issues around that.
Yeah, I don't think we specifically looked into that. We were relying on some statistics that we found at this called area health file. And that one, I think, has more general like clustering of the industries. So if there is anything that is related, any like statistic related to this chemical industry activity that you think is of great use, we can definitely incorporate that into our secondary analysis, because as I mentioned, our list of potential exposures can grow in the future and doesn't have to affect other analysis results we have. So we just haven't found a very good statistic at that time to use for this outcome.
Thank you. Yes, maybe we'll have an opportunity to talk afterwards. Thank you so much.
Sure, that would be great. Yeah, thank you.
Please join me in thanking Shanshan for an outstanding talk, and I'll turn things over to David Balshaw for the next speaker.
Thank you, Daryl, and thank you, Shanshan. That was a great talk, getting us started with some really exciting science, I think. So as we have been doing of late with Council, we wanted to take a deep dive into a topic. This was actually raised in our last Council with the concept that Fred Tyson presented. And so we planned a mini symposium to do a deep dive into epigenetics and epitranscriptomics and the crosstalk between nucleic acid modifications. So this was organized by Fred and Ashley Vargas, and we're going to turn it over to actually Trevor Archer, Deputy Director of NAEHS, to moderate our day. So Trevor.
Thank you, David, and good morning, everyone. So we really have a very exciting program that Fred and the team have put together. And so without further ado, I'll turn to Dr. Fred Tyson, who's the program director in the DERT and the Genes, Environment, and Health branch. And Fred is a leader at NAEHS and actually nationally, internationally on transcriptomics, epigenomics, and epigenetics. And so Fred will give us an introduction to the nucleic acid modifications portfolios that NAEHS currently supports. Fred, over to you.
Okay, thank you, Trevor, for that. And good morning, everyone. I feel really privileged to open up this session because it really features a roster of just superb scientists who are going to describe some of their lab's recent work in the areas of epigenomics, epitranscriptomics, or even multi-omics approaches. What I'm going to do in my brief time is give you a description or overview of how NAEHS support this aspect of our research portfolio has developed and continues to evolve. And to do that, let's start with a quick review of the central dogma. DNA is transcribed into RNA, which is then translated into protein. However, we know that this kind of straightforward linear example of the central dogma is not really what happens. So let's think of epigenomics as a process of turning on or off gene transcription with reversible chemical modifications that are heritable but do not alter underlying DNA sequences. And epigenomic marks like DNA methylation and histone modifications are recognized, installed, and removed by readers, writer, and eraser proteins. And other features of the epigenomic landscape are going to include chromatin accessibility, nucleosome positioning, and consideration of the roles of non-coding RNAs. So in mentioning RNAs, let me quickly switch over and give a working definition for epitranscriptomics. And we can think of that as a process of that are associated with the study of the reversible modification of transcripts or nucleosides, with the following caveat. The building block of alphabets, the RNA alphabet building blocks are not restricted to these 4 canonical bases. It's much more complex than that. And what you actually have are a number of different modifications that can be added to transcripts. And they are, again, like with epigenomics, they are installed, removed, recognized by reader, writers, erasers, proteins. When you add these chemical modifications to RNAs, you basically alter structure and function as a consequence of those modifications. Okay. I should also say that all RNA species that we've studied so far, we know are modified. That includes mRNAs, tRNAs, ribosomal RNAs, piRNAs, whatever RNA you think, it can be modified. And the other thing to keep in mind is also there are literally hundreds of these. The last count that we have from Modomics says that there are currently 300 modifications that are known. So having given these working definitions for epigenomics and epitranscriptomics, let's go ahead and do a quick overview of the portfolios. And the development of the environmental epigenetics portfolio really began with a meeting that was co-organized with Randy Girdle back in 1998 that brought together investigators to focus on genomic imprinting and environmental disease susceptibility. And what really ensued over the next 2 decades were a series of different workshops that made recommendations, and RFAs followed those. The RFAs were responded to, supported, and in between, this helped the portfolio grow over the last 2 decades. But the other thing that happened is when in between those RFAs being submitted, more unsolicited applications were submitted as well, reviewed well. And the result is we have really an epigenomics portfolio that touches virtually every portfolio that we support in the Division of Extramural Research and Training at NAEHS. And this investment in environmental epigenomics has been really fruitful, as evidenced by the increasing number of manuscripts that are published each year for this period that I'm showing between 2010, 20, and 2021 that have some aspect of environmental epigenomics in them and cite NAEHS as a source of funding. Now, at the same time we were investing in developing our environmental epigenomics portfolio at NAEHS, we were also able to co-lead the Roadmap Epigenomics program along with our sister institute, NIDA. And keep in mind, the Roadmap was the precursor to what's now known today as the Common Fund. The centerpiece of this Roadmap Epigenomic program was the Reference Epigenome Mapping Consortium, or REMCs. And the REMCs themselves had several notable achievements. Among them, they produced over 100 reference epigenomes from human ex vivo tissues and cells. Over 850 manuscripts were published. These were cited have been cited over 45,000 times. And it also gave rise to the formation and founding of the International Human Epigenome Consortium, or IHEC, which has set a lot of data standards and metrics that the field uses now. In fact, IHEC also has several data samples that people interrogate, and it really continues as an important component of the international epigenomics community. Now, working with these large consortia like a Roadmap and IHEC really kind of stimulated us to think about establishing similar types of programs and data sets closer to home. So that meant we were going to have to put an environmental exposure context in it. And to do that, we developed the multi-phase target program to leverage lessons that we learned with these larger programs. And target 1 focused on elucidating how epigenetic modulators respond to the environment and enhanced our understanding of how exposures perturb the epigenome. And when target 1 began, greater than 70% of the grants at NIEHS that were supporting epigenomics really had some type of exposure model and then interrogated impacts on DNA methylation. So the focus of this particular program, or a focus, was was to concentrate on epigenetic or epigenomic processes that were not DNA methylation. And thus we expanded the scope of work that NAEHS was supporting to consider histone modifications, readers, writers, erasers, chromatin accessibility, chromatin remodelers, nuclear architecture, as well as the role of non-coding RNAs, all with an exposure context. Now, the Target 2 program had a completely different goal. Okay, first, we wanted to determine if chemical exposures could generate specific epigenomic signatures in tissues that are associated with pathologies like metabolic or neurodevelopmental disorders. And if we could, would we also be able to identify correlative signatures in surrogate tissues that are less invasively obtained, like blood or skin or buccal cells? What type of information could be gleaned from epigenomic analyses of these surrogate tissues? And then the third goal was then to develop also a community data resource, much as the REMCs and IHEC do, so that the community would be able to ask their own questions, interrogate this data, and do this independent of the consortium. Okay, this consortium used next-gen sequencing approaches and looked at a diversity of epigenomic features. We used a wide array of exposures. I'm sorry, tributyl 10 was not listed on this figure, but we had a number of different exposures. We used an in utero mouse exposure paradigm and looked at a number of different adverse health outcomes. What I can tell you about this is there is a community resource that is available for people to interrogate, as well as there's an integrative analyses that have been done or are being done. They're currently in a state of being written up, so I don't want to really mention anything about them. You may hear about them a little bit later on in the day. What I do want to point out is, as I've told you before, we have a higher rate of publication coming from these programs. And I'm just showing you this slide, not going through any of these studies, but I also want to point out both of these programs have regularly published in high impact journals. So I want to move away from now epigenomics, switch gears and talk a little bit about our epitranscriptomics portfolio. And around the same time that the REMCs were generating human reference epigenome maps, there were groundbreaking discoveries made about the role of FTO or ALPBH5, which is an RNA demethylase, with M6A dynamics, and they were made by Chuan He. Around this same time, also Sami Jaffrey and Kate Meyer introduced a novel sequencing platform that allowed for base resolution mapping of this M6A transcript. And taken together, these two advances and discoveries really revolutionized the field of epitranscriptomics and catalyzed an explosive surge in research in this area. And this happened around 2010, 2011. Now, recognizing the potential impact of environmental toxicants on the deposition, removal, and recognition of RNA modifications and how this might be mechanistically associated with a number of different health outcomes, NIEHS embarked on developing a portfolio that would include exposures in an epitranscriptomics research. And we did this around 2017. Now, 7 years later, as a result of two modestly supported RFAs that were released in 2019, so we actually supported 5 R01s and 4 R21s, as well as having some effective sessions on environmental epitranscriptomics at several national societies, annual meetings. We were able to stimulate an increase in submission of applications that are unsolicited that come to NIEHS that address exposures in epitranscriptomics. And as a result, we're supporting a growing portfolio in this area that has right now over 35 active grants. Now, inasmuch as there are over 100 known human diseases with implications of perturbed epitranscriptomic processes, our portfolio of investigators are importantly looking at a diversity of health outcomes, different exposures, looking at a number of different readers, writers, and erasers of the epitranscriptomic marks, as well as looking at a number of different epitranscriptomic marks. They employ state-of-the-art technology and ask several great questions. What I want to point out here is there's really been remarkable productivity by our investigators in this portfolio. Again, you can see over the last few years, there's been a real escalation in number of publications that cite or mention epitranscriptomics with environmental context that are supported by NIEHS. And the other thing that I would point out is also routinely, these investigators are publishing in high impact journals. So what we have are people who are being prolific with their publication as well as discovery in terms of these things are going in high impact journals. So both of these portfolios are doing well in terms of productivity. And you might say, how are they going to advance and continue to contribute to the NIEHS mission? And I'm glad if you asked that question, I have an answer. It might not be the answer, but I have an answer. And that is, we need to recognize these different systems: genome, epigenome, epitranscriptome, proteome. These things do not exist in silos. They're all talking or communicating with one another just to carry out normal cellular regulatory activities. And this begs the question to me, is if exposures can come in and disrupt any of these communications that are going on between these systems or create new and appropriate communications. So there's really mounting evidence and data that shows crosstalks between the epigenome and epitranscriptome are occurring with great frequency. And this presents an opportunity to explore how exposures might contribute to disrupting existing crosstalks or even creating novel ones that have implications for pathogenesis. And to stimulate research in this area, we developed a concept clearance called EPCOT that was approved at the last meeting of this council in September. David briefly alluded to that a little bit earlier. We anticipate that these portfolios are going to really be able to contribute to the precision environmental health and exposomics initiatives that the NIEHS leadership has prioritized, and we expect that they're going to be able to contribute in many meaningful ways. So with that, I'm going to finish, well, I am finished talking about the portfolios in epigenomics and epitranscriptomics, and want to turn your attention to this great roster of speakers that we have coming up. I really don't have time to introduce them all, but I have great things to say about all of them. But we're going to start out with a presentation from Yen-Cheng Wang, who's at University of California, Riverside, that will speak using his multi-omics approaches to his research. We'll follow with a session that is developed or devoted mainly to epigenomics with presentations from Dr. Yvonne van Duve-Middendorf at the Van Andel Research Institute, as well as Justin Calcino, who's at the University of Michigan. And then we'll follow that with a presentation from Victor Cortez at Emory University. Victor actually uses both epigenomics and epitranscriptomics approaches in his study of BPA-induced transgenerational inheritance. So this will be an interesting talk to hear from as well. Following a brief lunch break, we'll hear from Tom Begley, who's at SUNY Albany. Tom has certainly been in our portfolio for a long time doing epitranscriptomics before we called it epitranscriptomics, and he'll talk about tRNA modifications in response to stress. We'll then follow up by a presentation from Dr. Yoon-Sun Nam, who's at University of Texas Southwestern. Yoon-Sun actually is not supported by NIEHS, but she's been a very strong contributor to our portfolio efforts for the last several years, generously donating her time, expertise, data, and stories of discovery from her group. And then we'll finish up with a presentation from Ting Wang, who's at Chair of Genetics at Wash U. Ting has, he's best known really for his work with transposable elements in human development and disease, but he's also known for his visionary work with data visualization platforms, as well as integrating and doing integrated analysis of multi-omics. So with that, I think I'm a little bit over my time. I'm going to stop and stop sharing my screen and turn the virtual podium back over to Trevor.
Thank you, Fred. And so Fred has generously taken the best part of my job and left me with the part of enforcing time and protocol for the rest of the symposium. But thank you, Fred, for a terrific introduction. And so just a quick reminder to our speakers, you've been allotted approximately 35 minutes. What we'd like to do is, if there is time within that space that we can have a few questions, we'll do that. And we want to really save the higher level discussion after you've heard all of the talks in the council discussion period at about 3 o'clock. That's specified there. So without further ado, our first presenter is Dr. Yinsheng Wang, who's a distinguished professor of chemistry at UC Riverside and uses a broad spectrum of multi-omic technologies. And I won't take any more of your time, Yinsheng. Welcome, and we look forward to your presentation.
Yeah, thank you. Shall I start the PowerPoint from my own computer, I assume, right? Yeah.
I think so, yes.
All right, yeah, thank you so much, Trevor, for the introduction, and also I would appreciate, the invitation from Fred, yeah, for speaking in this session. So today, I mainly talk about a project that's funded by NAEHS in the epitranscriptomic area. So overall, the research program in my lab, really covers many aspects of omics approach, including genomics, bioinformatics, and also proteomics, of course. So the emphasis of today's presentation will be on nucleotide repeat expansion and how RNA modification may contribute to the etiology of nucleotide repeat expansion diseases. So many of you probably have heard, like disease like Huntington's disease, yeah. So in these cases, basically, in the patient, they have the CAG, trinucleotide repeat expansion. So there have been many mechanisms proposed by this, but one very important observation is that, the disease onset, is negatively correlated with repeated length, whereas the disease severity is positively correlated with the repeated length. So for example, for Huntington's disease, generally speaking, if less than 34 repeats, people will not develop the disease. However, for anybody with a repeated length that's more than 40, so the disease penetrance is 100%. So in other words, there's a very sharp increase in disease incidence and also disease severity as the repeated lengths become longer. On the other hand, in even healthy humans, we all have this repeats. So the question is how? And there have been multiple mechanisms proposed about how the repeat expansion may give rise to disease development. So one mechanism is that, there's a repeat after expanded because they can bind to unique RNA binding proteins. So for example, the CUG repeat expansion observed in myotonic dystrophy, they can bind very strongly to a protein called muscleblind 1. So this protein is very crucial in splicing. So therefore, this sequestration of essential RNA binding proteins, is one of the mechanisms proposed for the disease etiology. Another mechanism has been shown is that, this toxic RNA, they can also be translated into proteins. So because the proteins translate from the repeat RNA, they are also highly repetitive in sequence. And as a result, they cannot fold appropriately in cells. So thereby generating proteotoxic stress. And importantly, this repeat RNA, they actually can translate in all three potential reading frames. So for example, the CAG repeat RNA, they can be translated in the CAG codon to generate polyglutamine. They can also be translated as AGC codon to generate polyalanine, or using the GCA codon to generate polyserine, and so on and so forth. So, but in our view, neither of these two mechanisms, on its own, perhaps cannot explain why there's a sharp transition in disease incidence and disease severity. So there must be some additional modifiers on top of this repeated length, that may give rise to what observed in the clinic. So as Fred alluded earlier, RNA, they can be modified, post-transcriptionally. So in the first project, I'm going to talk about, one of the RNA modification that's M1 methyladenosine, or M1A for short, that they can actually bind to TB43 and induce neurodegeneration. In the second project, I'm going to talk about, M6A, they can also occur in the CAG repeat expansion RNA. They actually can modulate the repeat associated non-AG translation. So anyway, so this one, I think most people are familiar with this slide. So we have the enzyme that can install the methyl group, for example, on either N6 position or the N1 position of the adenine base. There are also dedicated enzymes that can remove those methyl groups, from this methyl nucleosides. And then moreover, there are many cellular proteins, that can recognize the unique modified RNA nucleosides. So importantly, many of these eraser proteins, they are metallo enzymes. So for example, many of the demethylates, they're iron 2+, towards glutarate-dependent dioxinase. So therefore, many environmental explorers like cobalt and nickel, they may target those enzymes, thereby disrupting their activities. So likewise, many of the reader proteins, they are zinc finger proteins. So zinc finger proteins also are susceptible to displacement of the zinc with other toxic metals like arsenide and so on. So therefore, environmental explorer can target many aspects of the epitranscriptional program. So thereby, may perturb gene expression in cells and tissues. So our first hypothesis is that, because this, as Fred mentioned earlier, this modified nucleoside is prevalent, in essentially all types of RNA, right? We hypothesize that maybe the nucleosides in repeat expansion RNA, they can also be modified, and the modification frequency may be influenced by repeat length. So several years ago, we developed this LCMS platform that can allow us to detect a large number of modified nucleosides at the same time. Shown here, just the data for detecting N1 methyladenosine or N6 methyladenosine by using stable isotope level standard, using a technique called isotope dilution methodology. So with this, we started with a very simple experiment. So we internalized T cells, we transferred with a plasmid that can express CAG repeat of different lengths. So then we quantified the percent of the A that's modified to M1A or M6A. So you can see that the M1A modification frequency actually increased with repeat length, but M6A, you don't see that, right? And importantly, in typical mRNA, M6A level is present at a much higher level than M1A. But in this repeat RNA, especially with long repeats, like 38 repeats. So now if you look at absolute scale, the M1A level is actually exceeds that of M6A, right? So we also test this, in different animal models. So the first model we use is the Huntington's disease transgenic mouse model. So they either express shorter repeats with 7 CAG, or the disease relevant repeats with 140 repeats. So you can see that in striatum tissue, the longer the repeat, the higher level the M1A. But in cortex tissue, you don't see that. So this actually mirrors, what observed in the clinic, where in Huntington's disease, the part of the brain tissue that's impacted the most is striatum. Cortex is also impacted, but to a much lower degree. And also we have a Drosophila model. We collaborated with Nancy Bonini at UPenn. She has a Drosophila model of a disease called spinal cerebellar ataxia 3. So where they have the CAG repeat expansion as well. So you can see that a disease relevant expansion of 78 repeats confers much higher level of M1A. So likewise, we also use a C. elegans model. So in this case, we have a paneuronal expression of different lengths of a CAG repeat. So you again see that the longer the repeat, the higher level the M1A modification. M6A level, you don't see that, right? So we wonder, what's the mechanism that may contribute to repeat length dependent increase in M1A? So we reason that, likely, the repeat expansion RNA may modulate either the methyltransferase or the demethylase for M1A in the repeat RNA. So for the demethylation, as I alluded to earlier, they are primarily carried out by the iron-2 towards glutarate-dependent dioxinase. So they oxidize the methyl group to hydroxymethyl. So then they can undergo spontaneous loss of formaldehyde to generate methylate A. So we explored, the major enzyme for M1A demethylase and also the M6A demethylase. We asked the question, which one is involved in the demethylation of M1A in the CAG repeat RNA? So we found that only when we overexpress LKBG3, it can give rise to substantial decrease in M1A in the CAG repeat RNA, whereas the M6A demethylase or the other M1A demethylase has no impact. Yeah. So this really documents that LKBG3 is a major demethylase. So we also explore, which methyltransferase is involved in methylation of M1A in the CAG repeat RNA. So we knocked down all 3 known M1A methyltransferase. It turns out only when we knock down TRMD61A, it can give rise to substantial decrease in M1A in the CAG repeat RNA. In the meantime, M6A level is not affected. So then we examine, how the CAG repeat expression expression can affect the demethylase or the methyltransferase. It turns out that, the expression of LKBG3, the major demethylase, actually can be suppressed when we overexpress with the CAG repeat RNA. And the longer the repeat RNA, the lower level, the demethylase. We also check the methyltransferase where we do not see any change in expression level of the methyltransferase. So in other words, really the CAG repeat RNA can downregulate the eraser protein, thereby inducing a repeatedness dependent increase in M1A level. So this finding can also be recapitulated in mouse tissues. And interestingly, we only observe the decrease in LKB3 in striatum tissue. The cortex tissue, we do not see any difference. So this mirrors what we observe for the M1A level in these two types of tissues. So you may wonder why, how the CAG repeat expression RNA may suppress the expression of the demethylase. So in the literature, others reported that the CAG repeat expression RNA, they can be processed by dicer machinery to generate 21-mer small interference RNA comprised of the CAG repeat. And this has also been observed in Huntington's disease transgenic mouse model and the Huntington's disease human patients. And strikingly, if you look at the data here, the small interference RNA generated striatum is present at a much higher level than those in the cortex tissue. So then, we check the 3' UTR of the mRNA of LKB3, and turns out that in the 3' UTR region, there's a short stretch of sequence that's highly complementary to CAG repeat. So based on that, we hypothesize that maybe the CAG repeat generate small interference RNA, they can suppress the LKB3 mRNA through RNAi-like mechanism. So we test that by using a luciferase reporter. So we clone the 3' UTR of LKB3 to a luciferase reporter. So you can see that when we transfer the cells with synthetic RNA that's comprised of CAG repeat, or we overexpress CAG repeat RNA using a plasmid, in both cases, we can give rise to diminished expression of LKB3. And importantly, this decreased expression can be rescued by shutting down the RNAi machinery, by knocking down organon2. Organon2 is the catalytic machinery of the RNA-induced silencing complex. So if you knock down this organon2, you restore substantially the demethylase LKB3 expression. So mechanistically, the repeat length dependent increase in M1A is attributed to the CAG small interference RNA-induced silencing of LKB3, the demethylase enzyme. So with that, then as Fred mentioned earlier, there are many, proteins, that can recognize this modified nucleoside in RNA. So therefore, we hypothesize that the modified nucleosides in CAG repeat expansion RNA, they may also modulate their interactions with RNA binding proteins. So therefore, we use a quantitative proteomics approach. We did a protein-wide screening for M1A binding proteins. So in our data, revealed a number of candidate M1A binding proteins, and turns out that the top hit is a protein called TAR-DBP, also known as TDP43. So the mass spec data, sometimes nice. However, we are not sure whether the protein is a direct binder or we pull down the protein through indirect protein-protein interactions. So we have to validate this using purified protein. So by using purified protein in a synthetic RNA, we demonstrated that the TDP43 can bind to M1A with very high affinity. So the KD value is only about 18 nanomolar. And it can also bind to A or M60 containing RNA, but the binding affinity 2 to 3 times weaker. So therefore, this data show that TDP43 can bind directly and selectively to M1A containing RNA. So this protein, TDP43, is very important in the neurodegenerative disease field. There is a phenotype called TDP43 proteinopathy. This has been found in Huntington's disease and also in ALS and FTLD. So the key observations are, so normally TDP43 is a nuclear protein. However, in these diseases, the TDP43 become cytoplasmic redistributed and also form inclusion body. And they can also generate C-terminal truncation, and the protein can also be phosphorylated and ubiquitinated. So based on this, based on our proteomic data, and we reason that M1A modification in CAG repeat expansion RNA, they may enable RNA templated TDP43 protein aggregation, thereby leading to TDP43 proteinopathy. Because you have a very long stretch of RNA is heavily decorated with M1A. And the TDP43 is also known to be aggregation prone. So therefore, this RNA protein interaction may result in increased local concentration of TDP-43, thereby promoting this aggregation. So we test that, with a very simple experiment to begin with. So in cultured human cells, without ectopic expression of CAG repeat RNA, the protein is primarily nuclear localized. As soon as we overexpress CAG repeat RNA, the protein now becomes cytoplasmically distributed. And importantly, the longer repeat, give rise to larger aggregate size. And this can be restored, by overexpressing with the LKB3, the demethylase. So in other words, if we lower the level of mRNA in the repeat RNA, you can decrease the aggregate size of a TDP-43 protein. So we also examined, we also found that the TDP-43 protein foci actually co-localize with the CAG repeat RNA based on RNA fish experiment. And again, this co-localization can be decreased if we overexpress the demethylase LKB3. And also, we cannot observe this as effectively when we use RNA binding defective mutant of TDP-43. So in other words, this co-localization depends on the interaction between TDP-43 and the CAG repeat RNA. And also, it can be modulated by mRNA level in this repeat RNA. So we also modulate the mRNA level by downregulating the methyltransferase, tRNA61A. If we knock down, we also see diminished foci size of TDP-43 and a decrease in co-localization between TDP-43 and the CAG repeat RNA. By the way, this is using endogenous TDP-43, using an anti-TDP-43 antibody. So likewise, we also transfect the cells with synthetic RNA with 7 CAG repeats without M1A, or with 3 M1A, or with 16 repeats without M1A, or with 3 M1A. So you can again see that endogenous TDP-43 can be redistributed to the cytosol when we overexpress, especially the M1A containing CAG repeat RNA. And the longer the repeat, the higher level of M1A modification, the more cytoplasmic redistribution. And then we look at the second phenomena, TDP-43 truncation. We again see that in the detergent insoluble fraction, when we overexpress cells with the CAG repeat RNA, we can see more TDP-43 truncation. The longer the repeat, the higher the degree of TDP-43 truncation. And likewise, we can see this in Huntington's disease transgenic mouse brain. So in striatum tissue, we see higher level of the TDP-43 truncated protein in the disease relevant mice, but not in the healthy control. And we don't see this in the cortex. So this again parallels what we observed about the mRNA level in CAG repeat RNA and the protein expression level of the LKB3, the demethylase. So then we look at the dynamic property of the protein using assay called fluorescent recovery of the photobleaching. So this the method can allow us to determine how dynamic the protein is in cells. You can see that in cells, when we overexpress with long repeats of RNA CAG38, the recovery is only about 20%, even after close to 200 seconds. Whereas in the shorter repeats of CAG repeat RNA expression, the protein is quite dynamic, right? So with the CAG38 repeat RNA expression, if we co-express with LKB3, the demethylase, the protein becomes dynamic again. So in other words, the M1A in the CAG repeat RNA and its interaction with TDP-43 really renders the protein static in cells. So then we also look at the phase separation properties of the TDP-43 when expressing, when the cells express CAG22 repeat or 38 repeat. You can see that with the shorter repeats of CAG repeat RNA, when we add 1,6-hexane diol, this compound can disrupt phase separation by diminishing hydrophobic interactions. So you can see that with 22 repeats of CAG, the droplets can be readily dissolved. Whereas with the long repeats of 38 repeats, the droplet is static. It cannot be dissolved anymore, right? So in other words, the long repeats of CAG really converts a healthy phase separated droplets into a gel-like aggregates. So then we also look at this whether this interaction under the RNA modification has any pathological relevance. So we use C. elegans as a model. So in this case, we have a paneuronal expression of different lengths of CAG repeat. You can see that with 6-7 repeats of CAG, RNA expression, the neural cord become very discontinuous. We see a lot of gaps in the neural cords, yeah, and this can be quantified. So now in this background, if we had a heterologous expression of human LKB3, actually gaps can be closed again. And this phenomena cannot be observed with the catalytic dead version of the human LKB3. So similarly, we can use RNAi to knock down the worm ortholog of human tRNA61A. We can see that, with this knockdown, we also see that neural cord, we see more closure of the neural cord. So in other neurodegeneration can be partially restored in C. elegans. So likewise, we collaborate with Nancy Bonini, where they have this Drosophila model expressing 78 repeats of CAG. So in this background, if we ectopically express, human LKB3 protein, actually we can extend the lifespan of the Drosophila, whereas the catalytic dead enzyme cannot do that. And this, the mass spec measurement show that with the Y-type enzyme, we can reduce the level of M1A, whereas the catalytic enzyme, we cannot do that. And the M6A level is not affected by overexpressing human LKB3 as expected. So from this, we defined really the reader, writer, and eraser proteins for M1A in CAG repeat RNA. We also demonstrated that, the CAG repeat RNA, the longer the repeat length, the higher level the M1A. And mechanistically, this is due to the CAG repeat RNA process small interference RNA. They can silence the key demethylates for M1A in CAG repeat RNA, thereby stimulating the M1A formation in the long repeat RNA. We also found that, this M1A and CAG repeat RNA, they can bind directly and strongly with the TDP43, thereby changing the phase separation property of a TDP43 protein. So normally the TDP43, has a physiologically relevant phase separation. In other, the protein can get in and out of the phase separate properties very easily. However, with very long repeats, because a high level modification of M1A in the repeat RNA, they convert the healthy phase separation into a pathological gel-like aggregates. Yeah. So now, we switch the gear to the next part of the project, where we show that another modification, M6A, actually can mechanistically contribute to repeat associated non-AUG translation from CAG repeat RNA. So as I mentioned earlier, the CAG repeat RNA expansion, they can be translated in cells into all three potential reading frames to generate, polyglutamine, polyserine, and polyalanine. But the mechanism, how this repeat RNA can be translated without initiating codon, is not clear. And also, I should mention that, this kind of repeat associated non-AUG translation not only happen in CAG repeat expansion diseases, but also occur in other diseases like G4C2 repeat expansion as observed in ALS, right? So earlier work, by Sammy Jeffrey showed that, under stress conditions, especially on heat shock, actually some mRNA, especially heat shock protein mRNA, they can be translated in a process known as cap independent translation. So in other, M6A can be induced in the 5' untranslated region of the messenger RNA of HSP genes, thereby inducing translation without relying the 5' cap structure. So since we can detect also M6A in the CAG repeat RNA, so we reason that M6A in the CAG repeat expansion RNA may promote this repeat associated non-AUG translation through a similar mechanism as cap independent translation. So this can be tested, if we can modulate the M6A level by genetically change, the methyltransferase or demethylase level in cells. So we did that by using a construct, this can allow us to monitor directly the RNA translation, in all three reading frames by using three different tags, where the MIG tag monitor polyQ generation, and the FLAG tag monitor polyserine generation, and the HA tag monitor polyalanine generation. You can see that you have 293 T cells. If we knock out LKB3, the RAN translation product from all three reading frames are decreased substantially. And this can be restored by re-expressing Y-type human LKB3 but not the catalytic dead mutant. So in other words, after knocking out the methyltransferase for M6A generation, we can shut down the RAN translation. And this is done using Western blot. And similarly, if we inhibit META3 using a small molecule inhibitor, the phenomena is very, very similar. We see substantial decrease in RAN translation from all three different reading frames. And this can also be monitored by fluorescent microscopy, where the polyQ is tagged with the GFP. So we can see that after we knock out META3, we see substantial decrease in GFP signal. And this again can be restored by re-expressing wild-type human META3, where the catalytic dead META3 cannot do that. And similarly, for the polyalanine reading frame, we also see the same phenomena. So likewise, for polyserine, in this case, interestingly, polyserine actually form foci, in the cytosol. So you see that after knockout META3, this foci of polyserine is just almost disappeared. And this again can be restored with ectopic expression of Y-type META3 in the knockout background. And the catalytic dead enzyme cannot restore that. We also test this by knocking out the demethylase. We found that in the CAG repeat RNA, FTO is a major demethylase for M6A. You can see that if we knock out FTO, we again see pronounced increase now in the RAN translation product. So together, we found that the RAN translation from CAG repeat RNA can occur through a very similar mechanism as a cap independent translation, and it can be promoted by M6A in CAG repeat RNA. And I think it's important also to examine whether RNA modifications also contributed to RAN translation from other repeat expansion RNA, like G4C2 repeat RNA. And this going back to this mechanistic slide, I think this RNA modification really contribute to many aspects of the proposed mechanism. So first of all, we found that the CAG repeat expansion RNA, they can sequester TDP43. That's essential splicing related enzyme related proteins, And importantly, this sequestration also gives rise to aggregate formation of TDP43. So in other words, this TDP43 the CAG repeat expansion induced protein aggregation not only can be generated from the protein product translated directly from the repeat RNA, it can also be generated for other RNA binding proteins. And also we demonstrate that M6A also play a very crucial role in modulating this RAN translation from the repeat RNA. So with that, I want to thank my co-workers, primarily my postdoc Yu Xiangsun, who spearhead all this research efforts for both projects, and also assisted by Hui Dai, who helped with the C. elegans part of the research. And the C. elegans part of work is done through collaboration with Professor Weifeng Gu at the UC Riverside, and the Huntington disease mouse studies done through collaboration with Professor William Yang at the UCLA, and the Drosophila work is done through collaboration with Professor Bonini at the UPenn. And with that, I want to thank all my co-workers, and I also want to thank NIEHS for supporting this line of research. And I want to thank you for your attention, and I'd be happy to take any questions.
Yinjing, thank you very much. That was a terrific talk, very beautiful work. We are a little bit behind, but we have a couple of questions. Karen, if you'd like to start.
Yeah, Yinjing, that was really great. Thank you for all that mechanistic understanding at the RNA level. But I have a question about, so clearly your results on the disease represents with longer repeats, right? So that starts at the DNA, right? So the DNA expands. So in your model, I would propose that you put the DNA in a hairpin as well. So we know that those CAG repeats form hairpins and are processed aberrantly, leading to expansions and often deletions as well. But nevertheless, my question is, so while it's rare, adenine in humans can also, at the DNA level, can also be methylated. And do you know if methylation of adenine in the CAG repeats impacts expansion? Because, right, if the DNA is not, the other question I have always wondered is why is it that particular level, right? Why after 40 repeats it's a problem, but it's not at 32? And you show some repeat dependent results with your protein bindings and interactions on the RNA. Do you have an explanation for the repeat length requirement for protein interaction, and also the question about the DNA, because if we could prevent the expansion at the DNA level, we wouldn't have to worry about the RNA and protein, right?
Yeah, that's a great question. Yeah, this I mean the mechanism underlying the repeat expansion is actually highly associated with the DNA repair, especially the mismatch repair machinery, MSH2 and MH3, as you know, is essential for repeat expansion in both human population and also in animal models. Yeah. So that's one thing. So M6A, whether N6 methyladenine is found in repeated containing DNA, that's a very good question. We have not looked into that. Certainly, it would be interesting to look into. Yeah. So from our data, certainly the longer the CAG repeat, the more pronounced decrease we see for the demethylase. I think perhaps the longer repeat, you have a higher likelihood to be processed into small interference RNA. So therefore you can silence
that's
better.
What I think. Okay, so the processing is more efficient at longer lengths.
Exactly. Yeah. So therefore you can shut down the demethylase ARKB3 more effectively. So that's, I think, is one of the mechanistic aspects.
Yeah. Very interesting. Great work, and Cheng. Thank you.
Oh, thank you, Karen. I appreciate that. Yeah.
We'll take one last question from Dr. Begley.
Hi, Yang Cheng. That was a wonderful talk.
Well, thank you, Tom.
Really beautiful work. So any insight on, when the methylation of the RNA is occurring? Is it during like an R loop formation, or what about also any insight on sort of modifications of the Cs in those repeats too?
That's a great question. Yeah. So certainly ARKB3 can also demethylate M3C. Yeah, that's something we are actively exploring at the moment. Yeah. So what's the second question?
I mean, it was about some of the R loops, but
yeah, we don't know where it happens. Yeah, that's sorry, that's a good question. Yeah, there's no right now. Certainly we have idea how to interrogate that. We certainly will be very interested in looking into it. Yeah. So in other words, whether this happens inside a nuclei, modified first, then delivered to cytosol, then recruit TB43 there, right? Or it happens in a cytosol, then you recruit TB43 there. The temporal aspect, yeah, we don't know. That's certainly very important to explore. Good question. Yeah, thank you.
Yeah. Thank you, Dr. Wang. And as I mentioned earlier, we will have time during the council discussion for further discussion of these talks. So let's move on to our next presentation, which is by Dr. Yvonne Fundev-Mittendorf from the Van Andel Research Institute, where she's a professor of epigenetics. Yvonne is an internationally recognized expert in chromatin biology and metal toxicology, and she'll tell us about hopefully some of her more recent work on arsenic, lung cancer, and perhaps the involvement of circular RNAs. So without further ado, Yvonne, please.
Okay. Thank you so much for inviting me here today to talk about some of the work that we do in the lab. And it's mainly trying to understand how the epigenomics reprogrammed in response to environmental toxicant. And the toxicant we use is environmental arsenic exposure. And you're probably wondering, why should I care anyway? It's been linked to cardiovascular diseases, diabetes, cancers, including other diseases. And you might say, well, where do we get it? Contaminated water, contaminated food, and occupational hazards. And you might still say, well, it still doesn't matter to me because I'm always clean. But this is what the New York Times actually put last year, where due to climate changes in climate, water that typically will cover the bed, the soil underneath the water, now that water dries up, and you have arsenic that is found in the soil, and that becomes exposed to the elements, and it can actually go, so many more people can be exposed that way through wind blowing it to thousands and thousands of miles away. And so they actually claim that it's potentially an environmental nuclear bomb. And so the question for us, we've always wondered, how does arsenic cause carcinogenesis? Typically, I'm just showing you here in Michigan, where in some places in the well water where I live, Kent County, you have about 0.5 micromolar of arsenic present there. But in other regions, you could have equal as high as 220 microgram per liter. It's been shown that in low concentrations, arsenic does not directly mutate DNA. So how could it be driving carcinogenesis? And in the lab, we hypothesized that it could be doing that through epigenetics, changing the chromatin structure, thereby to drive cancer. And so this is our working hypothesis that arsenic would directly or indirectly change the chromatin structure, which would change gene expression and lead to cancer. And so we decided to build a model where we're looking at how arsenic in cells could actually drive carcinogenesis. And so we treat Bs2B epithelial lung cells. We treat them with 0.5 micromolar of arsenic, which is typically what you find in the well water in Michigan and in many other places as well. We treat them with 0.5 micromolar of arsenic, and we watch weekly progression of the cells towards carcinogenesis. We see that the cells become more migratory and eventually they revert. When they revert, they actually are able to form colonies in soft agar, and indeed also they're able to form tumors in mouse xenograft. What we've seen is that with gene expression analysis is that the cells move from epithelial to mesenchymal and they revert to epithelial. The epithelial here is not the same epithelial here. And indeed in cancer, it's been shown that typically cells will become more migratory and they become more mesenchymal. Once they find a region where they want to bind to, they become more, where they want to colonize, they become more invasive and they revert towards the epithelial state. So we think we have a model to begin to understand how arsenic might directly or indirectly change the epigenome to drive cancer. And so I'll talk to you about 3 projects, and we're indeed very, very grateful to the NAEHS for funding us through these projects. One, I'll talk to you how arsenic could directly target the 3D genome rewiring. I'll also talk to you about how it could change histone, incorporation of histone variants into the chromatin, and that will change gene expression. And lastly, I'll talk to you about a circular RNA that also comes out from a gene that is important in regulating chromatin structure, and that how could also drive carcinogenesis. So when we started this project, we did like everyone else, where we profiled the epigenomic changes in arsenic transformed cells. And we thought, typically people will talk about in arsenic transformed cells that you have hypomethylation. When we did that analysis, RNA methylation sequencing, we saw both hypomethylation changes as well as hypermethylation changes. We think we can explain hypo, but we don't as yet know how to explain hypermethylation. And so we think it's actually a reprogramming of the methylation status. But for hypomethylation, what we noticed was that the expression of DNMTs was low in arsenic transformed cells. So we said, possibly this is how you're having hypomethylation. But the question is, how is arsenic directly targeting hypomethylation or the expression of these DNMTs? And lo and behold, when we looked at the promoters of DNMTs, we saw a binding site for a protein, CTCF, which is one of only 2 3D genome organizers, changing your 1D chromatin structure into a 3D genome architecture. We saw that in normal cells, CTCF is able to bind. In arsenic exposed cells, we see CTCF not binding, and we assume because that CTCF is no longer binding that we're having a decrease in DNMT expression. Interestingly, the question is, how is arsenic then targeting CTCF? It's been shown that proteins that have zinc fingers, especially those that have 3 cystidines and 1 histidine coordinating the zinc ion, or 4 cystidines coordinating the zinc ion, arsenic can come in, displace that zinc, insert itself into it, and that protein will no longer be able to function properly. Interestingly, CTCF has 11 zinc fingers. 10 of them are C2H2, which should not be a target of arsenic. The 11th zinc finger is C3H1. So the question for us was, is arsenic directly targeting CTCF by targeting this 11th zinc finger? So we also have shown in vivo, and I'm just using a summary here, where we see in normal cells, and this occurred mainly with tumor suppressors, where we see a weak CTCF binding at the proximal site, and we see that in normal cells, CTCF is binding. And then when we treated the cells or exposed the cells to arsenic, we see that CTCF is no longer binding. And our working hypothesis here is that you're probably having a double whammy where this weak CTCF binding site, CTCF doesn't like to bind. It probably requires all its zinc fingers to help it bind to that site. But you now have arsenic inhibiting this 11th zinc finger, and so CTCF is no longer able to bind. So this is when we looked at just those that have single weak CTCF binding sites. Those that had dual CTCF binding sites, and we saw this mainly with oncogenes, we saw them there was a weak CTCF binding site to the proximal to the promoter, and a strong distal enhancer CTCF binding site. In normal cells, we saw CTCF binding, and we think it's actually looping over to interact with CTCF to drive gene expression. However, now when we expose cells to arsenic, we see that CTCF is no longer able to bind to the weak CTCF binding site, but to the strong CTCF binding site, we saw a really enhanced occupancy of CTCF to that site. We think again that most likely the CTCF is still looping around and holding onto this promoter to enhance gene expression. Is this true that it's targeting the 11th zinc finger? Sorry, before I go into that. So we did also a gel shift analysis where we took a CTCF we purified CTCF recombinantly. We also purified strong CTCF binding sites as well as weak CTCF binding sites. What we noticed was that if we do gel shift and assays, we see CTCF binding really nicely to the strong CTCF binding sites and causing a gel shift. And to the weak CTCF binding site, CTCF doesn't like to bind. We took that CTCF now, dialyzed it in a buffer that has arsenic with the aim of displacing arsenic from that zinc from that 11th zinc finger. When we did those analyses, lo and behold, we do see that it's still binding to the strong CTCF binding site. There is a decrease, but now to the weak CTCF binding site, we don't even see binding at all. So we decided to ask the question, is it really targeting the 11th zinc finger? And so again, this is the 11th zinc finger, that is C3H1. And so we mutated that to C2H2. When we did that analysis, in the absence of CTCF or in the presence, in the absence of arsenic or the presence of arsenic, we see that to the strong CTCF binding site, it's binding really nicely. There was no impact on that. Now to the weak CTCF binding site, it's even more interesting that to the weak CTCF binding site now, with that change in the 11th zinc finger, it's really binding nicely, and also in the absence or presence of arsenic. And so I just give you a table here showing you the EC50 values to summarize what I've just shown you. So in a wild type, CTCF can bind to the strong CTCF binding site. We see about 51 nanomolar being used. To the weak CTCF binding site, it doesn't like to bind. If we take that CTCF, dialyze it in a buffer that has arsenic, we now see that it moves it even to the strong, it reduces the binding to the strong CTCF binding site, but to the weak CTCF binding site, it actually abolishes binding. If we mutate that CTCF, we see that there's really no change in the absence or presence of arsenic, but now to the weak CTCF binding site, it makes it even a better binder, and we don't see a difference between whether arsenic is present or arsenic is not present. So we do think that that's the 11th zinc finger that is targeted. So what does CTCF do? And so this is typically a double helix. You beat on the strain form of chromatin. And CTCF, you typically have this 30 nanometer chromatin structure looping around where it brings enhancer close to the promoter, and CTCF is holding onto this basis. And you could have those loops folding onto each other to form topological associated domains. And CTCF is also holding onto this region. CTCF in that case could act as an insulator, blocking the repressed topological associated effects from moving into the active topological associated domains, such that when they fold onto each other, you have A and B compartments. And if you stain your chromosome, they stain according to territories based on the transcriptional activity. So our working hypothesis is that CTCF interaction holds onto this loop. It blocks this enhancer from ectopically enhancing gene expression from this black promoter. But if you now disrupt CTCF, you now have a bigger loop that is formed, and you're having this enhancer now close, possibly close to this promoter in three-dimensional space, so that it can actually be driving gene expression now from this promoter. And if that's an oncogene, you could actually drive gene expression that way. We've now worked with Hi-C, where we're measuring the interaction maps. And if we look at how, if we start from the non-treated cells and we're going through the weekly exposure to arsenic and actually transformation, we see, I just show you examples here where we see this interaction strength right here, where you see the squares a lot stronger. And as you move through the weekly exposure of arsenic and transformation, you're actually seeing a difference in the interaction strength between this compartments. And also we've seen compartment switching where in green is the active compartment, in red is the repressed compartment. And I show you an example here where you see in this red repressed compartment, you see this green compartment showing up. But as you go through the weekly exposure, you now see a compartment switch where most of the active ones have now switched into the repressed ones. And I'll just summarize this for you in case you don't believe the subtle changes, in that if we look at eigenvector measuring a compartment interaction with a compartment, we see a strength of about 5. And as we move down, it reduces to about 3. If we look at the B compartment and D compartment, their interaction, it starts with about 8 and it moves down to about 4.3. So suggesting to us that most likely arsenic mediating CTCF binding is possibly changing the interaction between the compartments and even bringing enhancers close to promoters. We're now sequencing in depth to actually look at the enhancer to promoter interaction strength. And we're doing this an analysis together with Professor Decker at UMass. We also now have cell lines where we've mutated the 11 zinc finger of CTCF to C2H2, and we're seeing also that the transformation potential is a lot slower. I talked to you about zinc. Is this all about zinc? We're trying to think about zinc as a therapy or therapeutic. And so we've also started treating cells with zinc. We see the effect of zinc a lot stronger in reducing transformation compared to CTCF mutation. So we think maybe in understanding how this might be impacted by zinc or how arsenic is displacing zinc in many other proteins might actually help us to see whether we can use zinc as a therapy. So I've talked to you briefly about the 3D genome rewiring. I'll also talk to you that we did see, which was very exciting, we saw also incorporation of histone H2B variants. And when we started this analysis, we looked at, again, as we start with everything, we profiled all the various histone variants that are present in arsenic transformed cells. And when this data came forth, it was kind of very confusing to me because I'd never heard of this histone H2B variants. Typically, people talk about H2A variants and H3 variants. H2 variants have received very little attention. And we saw differential expression of this H2B variants. And so our working hypothesis for that is that canonical histones, or why are these histone variants coming up? Our working hypothesis is that canonical histones have a stem loop. Their transcripts have a stem loop at the end of their at the end of the transcripts, and typically are bound by the stem loop binding protein. We and others have shown that arsenic can come in and degrade that stem loop binding protein. And if it does that, our working hypothesis then is that the canonical transcripts, their expression or the stability in the cell would be lower. And so we think the cells are then, because they have the same amount of DNA and they're trying to compact their DNA, they now make histone variants that have a polyadenylated tail. And so we think it's a compensatory mechanism for that. But interestingly, when that happens, you can imagine that a histone variant comes into the nucleosome. When it inserts itself into it, it could actually change properties of that nucleosome. It could change how that nucleosome breathes, which means the DNA that opens and closes around the nucleosome, how that opens, how fast that opens. It can actually change the stability of the nucleosome, or it can even change the modifications that are present on the nucleosome, which means that you have different readers and writers and erasers coming in, and that would also then change gene expression that way, which is typically transcription factor accessibility. Interestingly, This project has been a little difficult, but we're getting heads on it because these histone variants differ by 1 to maximally 3 amino acids. And so you can look at this as a canonical histone H2B. Histone B differs by just this serine, isoleucine, and this isoleucine at position 40. H2B differs by just this threonine. H2BJ differs by this isoleucine and this alanine, and K differs by just this alanine at the C terminal tail. And so the question is that, then we looked at also their expression in cancers. And this was kind of really exciting to see that the expression in cancers are different, and also they're also cell type specific. And even making more interesting is that the mutational landscape of this histone variants are very different compared to the canonical. So this is a canonical mutational landscape in tumors. And we see that B differs by this R92 and E133K, whereas K also differs by different amino acids that are mutated, J and likewise D. So the question is that how can a single amino acid change how this histone variants are expressed, and how could they change the mutational landscape? And so this has led to the field of oncohistone, where it's been shown that mutations of other histone variants actually drive specific tumors. For instance, H3.3 G34 mutations have been shown to promote aberrant PRC2 activity and drive tumor progression that way. And the late David Alice, who was actually the pioneer in histone modification, says every amino acid in a histone counts. And they've also shown how histone variants could actually lead to mammalian development and disease. So in the absence of us not having the ability, we've worked for about 2 years now with Thermo Fisher trying to get antibodies that would specifically target histone variants, H2B variants, and we've not been able to get that. But I'll explain, we've kind of used another system to understand how this variants could actually be driving gene expression. So what we did is we did biophysical assays where we labeled the 601 146 base pair DNA with Cy3. We labeled the histone octamer with Cy5. And then the idea is that if Cy3 and Cy5 come close to each other, you'll have a FRET signal from Cy3 transferring onto Cy5. And you'll be able to read the FRET signal from Cy5. And we also put a GAL4 binding site to the beginning of the nucleosome. And indeed, this is the region where it's been shown that most transcription factor binding sites are present within the nucleosome. And so we're measuring the rate of nucleosome breathing with the various histone variants. And when we did that, this was very exciting to see that if we look at H2BC compared to H2BD, H2BC is the canonical histone as shown here. H2B differs just by this threonine here. We see that we did not see any change in the amount of GAL4 that is needed to open the nucleosome. We see 3 nanomolar of GAL4 is needed to open H2BC compared to H2BD. But when we look at H2BK, it seems to require about twice the amount of GAL4 to open up the nucleosome for H2BK. H2BK differs by just this alanine right here. On the other hand, now, if we look at H2BJ, which differs by the alanine to the canonical, as well as this isoleucine, we see that it requires 3 times the amount of GAL4, 3 nanomolar with the canonical histone, as opposed to 11 nanomolar for H2BJ. It requires so much more to open up the nucleosome, suggesting to us that it seems as if H2BK and H2BJ seems to be holding the nucleosome in a more closed conformation. To answer that, we then did other assays where we had the linker DNA added now to the 146 base pair fragment. And so there are about 50 base pairs of linkers. And so what we're looking at is if we assume that the variants are causing the nucleosome to close up, is it possible that they're also changing the trajectory of the linker DNA that gets out of these nucleosomes? So you could imagine that you could have one conformation where the linker DNA moves right out this way, or the other way where the linker DNA folds over onto the histone, onto the nucleosome. And so we're doing this assay testing with micrococcal nucleus. And this was very, very, very exciting to see that. When we looked at H2BC, which is the canonical histone, with the same amount of DNA, as well as the same amount of enzyme, micrococcal nucleus, we see that the H2BC was readily digested, whereas for H2BJ and H2BK, they were slower in digestion, suggesting to us that it's most likely that that linker DNA folds over onto the H2BJ and H2BK. And so this is a summary of what we're seeing. If we look at the histone variants that differ at the N-terminal tail, we don't really see a difference with how the linker DNA is folding over. And that's what we saw also with the FRET, where we did not see a change in the nucleosome breathing. However, with those histone variants that have a C-terminal tail change, as well as at position 40, the isoleucine, we see that it takes a longer time to digest the linker DNA, suggesting that that linker DNA possibly is folding much over and closer to the nucleosome. We also know that in cells, the nucleosomes are not just single. And so we asked the question, maybe the subtle changes, we might be able to see them even nicer if we use a chromatin array. And so we reconstituted nucleosomes on a 17-mer nucleosome array, and we use atomic force microscopy to actually measure how compact these arrays are with the histone variants. And I'm just showing you an example here for H2BJ because the other ones, again, look very similar to this one. If we measure the height of each nucleosome within that array, we did not see a difference with the nucleosome height. But interestingly, when we measure the diameter, it showed that H2BJ was even smaller. The diameter was smaller, suggesting again and confirming with what we've seen that the DNA is probably holding tighter to the nucleosome. We also, this also translated to the nucleosome volume, where we see it also smaller compared to H2BC. Likewise, if we measured the array now, the 17-mer array, we did not see a difference in the height, but then to the diameter, we see that in H2BJ, the diameter is now bigger. And so if you could imagine that the 30 nanometer chromatin structure and the plane of a 30 nanometer chromatin structure, and you could see several nucleosomes on that 30 nanometer chromatin structure. If you have a looser or a bigger, sorry, let me go back here. If you have a bigger diameter and volume, it would mean that less nucleosomes are able to fit into that 30 nanometer chromatin structure. But if you have a tighter one, it will mean that more nucleosomes are able to fit into that plane of that 30 nanometer chromatin structure. And indeed, that's what we're seeing. So what I've shown you so far to summarize this one is that we do not see this variance impacting nucleosome stability. We see this variance impacting nucleosome breathing, possibly facilitating transcription factor accessibility. And it's not only a single nucleosome, but this translates also to a nucleosomal array. And so we're now working with we now have cells where this variants are tagged, CRISPR-Cas9 tagged, and we're trying to understand if this is true in vivo. Do they drive carcinogenesis? Where are they located during arsenic-mediated carcinogenesis? What are their PTMs, interactomes? And this is exciting because while everybody talks about H3 variants, H2A variants, H2B variants have just not been studied. And I think they've not been studied because people thought it's just one amino acid that is changing and might not even have an effect to that, but we're actually seeing that effect. And I think it's opening up a field that is very important in biology as well as in toxicology. So lastly, I've talked to you about chromatin changes, how the 3D genome rewiring could be changed, how we also are seeing histone B variants also changing and incorporating into the chromatin structure. And that would lead all to changes in gene expression. Just like every other person, we measure gene expression as transcript abundance or alternative splicing. But one area that has received very little attention is back splicing. And I'll talk to you about a circular RNA that is coming from SADB2, which is also a chromatin organizer, and that could play a role in arsenic-mediated carcinogenesis. So before I go on, what is back splicing? So typically this is your pre-mRNA. And so when you splice, your mature mRNA would look most likely like this, where you have your 5' cap and your polyadenylated tail. For back splicing, you could have exon 4 back splicing onto exon 3, and you have this circle here. And the only way you'd be able to determine this is this unique back splicing. Black splice junction. And most of the time people had thrown this away because they thought it was junk RNA. And the reason for that is that they're expressed in very low amounts. And so you need a strength, bioinformatic strength, to actually be able to determine this back splicing junction. And you also have to sequence your RNA in high depth to be able to capture this back splice junction. So what do circular RNAs do? People have shown that circular RNAs could actually regulate the rate of RNA polymerase if they are found in the nucleus. People have also shown that it could act as microRNA sponge or decoys. It could actually sequester RNA binding proteins. And recently, people have shown that it's been translated. I'll tell you about a circular RNA that we found that's been translated. We also, again, we start our projects by looking at all profiling, just major profiling. What were the circular RNAs that were translated in arsenic-mediated carcinogenesis? And when we did that study, we saw lots of circular RNAs that were dysregulated, but we wanted to confirm with nanostrain. And so we used a panel. Is it only the circular RNA that's coming from the host gene, or is the linear mRNA also coming from that host gene? And lo and behold, when we did that, the most dysregulated in our cells was SADB2, a linear mRNA, which was highly expressed, but there was also a circular RNA coming from SADB2 that was highly expressed. SADB2 has been shown recently to play a role in anchorage-independent growth and cell migration in human bronchial epithelial cells. They've also shown that it plays a crucial role in arsenic-mediated carcinogenesis, and they've suggested that a MER31 could be regulating SADB2 linear mRNA expression. And also recently, there was a paper that showed that a circular RNA also emanating from SADB2, which is different from our own circular RNA, circ8928, plays a role in the progression of non-small cell lung cancer. And so we were asking the question, could this circular RNA be playing a role in arsenic-mediated carcinogenesis? But before I go on, I'll just tell you a little bit of what SADB2 does. So again, your double helix beats on a string form of chromatin, your loops, A and B compartments, and this is your chromosome. This is your nucleus. SADB2 plays a role as a chromatin organizer by bringing different regions of the DNA. It actually binds to AT-rich regions, MAR-associated regions of the DNA, and brings it to the nuclear matrix. And so in that case, you could imagine that if it's oligomerizing or tetramerizing, it could bring several regions of the DNA to the nuclear matrix. And by doing so, could actually bring an enhancer close to a promoter and drive gene expression that way. So we asked the question, we're seeing both circular RNA coming up as well as the linear mRNA coming up. Is it because of transformation, or is it because of arsenic exposure? So to answer that question, again, this circular RNA comes from exon 3 to 6 of SADB2 mRNA. And we asked the question using qRT-PCR whether this circular RNA is as a result of transformation, or is it due to exposure with arsenic? And so we acutely exposed cells for 48 hours with different concentrations of arsenic. And it was very interesting to see that SADB2 linear mRNA went up, as well as the circular RNA went up. We also were able to show it in our model of arsenic, weekly exposure of arsenic to transformation, where we see that as the SADB2 linear mRNA is going up, so also is the circular RNA going up. And at first we were perplexed with this. Why are they co-expressed? Is one controlling the other, or how are they expressed? And I think we got the answer when we did this experiment. When we knocked down SADB2 and measured SADB2 expression, we see SADB2 go down, but the circular RNA did not go down. On the other hand, if we knock down the circular RNA as expected went down, and SADB2 now went down, suggesting to us that is it possible that the circular RNA is coming in to protect the SADB2 linear mRNA? And we think we got the answer from this experiment. So if we overexpress the circular RNA by itself, it went up. SADB2 did not go up. But if we overexpress SADB2 now, the circular RNA went up. Again, suggesting to us that it seems as if every time SADB2 is there, the circular RNA is coming up, possibly protecting SADB2 RNA from degradation. Indeed, when we knock down both the circular and SADB2, remember that if you're knocking down the circular, also SADB2 goes down. We see inhibition of cell proliferation, and we see also inhibition of cell migration. If we overexpress the circular RNA and SADB2, we see them also increasing cell proliferation as well as cell migration. And indeed, we then did RNA-seq. What was very exciting for us is that we saw that there was a pathway that was targeted by SADB2. When we overexpress SADB2, there was a KRAS pathway that was targeted. Indeed, when we looked at arsenic transformed cells and the genes that are targeted in these KRAS pathways, they were very similar or almost similar to the same genes that were upregulated when SADB2 was also overexpressed. Similarly, for those genes that were downregulated, KRAS pathways targeted genes that were downregulated in arsenic transformed cells, those genes were also downregulated in SADB2 overexpressed cells. And I just show you an example where it just made our lives so much easier and exciting, in the sense that typically people think of KRAS as a mutation, but could we also have diseases that are actually targeted by epigenetic changes in KRAS pathways? And so this is what I just showed you earlier, where we in arsenic transformed cells, we see those genes that are targeted by in KRAS pathways that are targeted by arsenic transformation were also targeted by SADB2 overexpression. And we see that those genes are overexpressed similarly, and those genes when they're downregulated, they're also downregulated similarly. But then we now knock down SADB2 or knock down the circular RNA. And lo and behold, those genes that were upregulated in arsenic transformed cells and also upregulated in SADB2 overexpressed cells were now downregulated, seemingly to reverse that condition that we're seeing. Likewise, for those genes that were downregulated, they now become overexpressed in SADB2 overexpression, and the circular RNA knockdown as well. And I just show you a summary of that aspect, where those genes that were upregulated in arsenic transformed cells were also enriched in SADB2 overexpressed cells, and those genes that were downregulated were also downregulated in arsenic transformed cells. Likewise, if we knock down SADB2, those genes that were previously up in arsenic transformed cells were now downregulated, and those genes that were previously downregulated are now overexpressed. And we think this is actually chromatin regulated, because due to time, I'm not showing you that, but we see a very similar pattern in ATAC6, suggesting to us that most likely SADB2 is changing the chromatin structure to drive this gene expression patterns. So to summarize this aspect, we see that the circular SADB2 seems to be protecting the SADB2 transcript. Is it doing this via sponging microRNAs? We seem to have targets that we're analyzing right now to see whether it's sponging microRNAs and thereby allowing SADB2 to be expressed. We also see that SADB2 overexpression targets similar KRAS pathways as in arsenic transformation. And just to finish, we got this really exciting data that I thought I'll share, because I told you previously that we think this circular RNA is translated. Most of the time, people throw it as either junk or they say it's non-coding. So again, if we look at the transcript of SADB2, it starts from 1 through 11. And you have if the peptide for SADB2 protein, you have the ubiquitin-like domain, the cut-like domain, and it also has the MA-associated, the region it uses to bind DNA, which would be from exon 7 through to exon 11. This circular RNA comes from exon 6 through to exon 3, and so it has a start site and it has a nuclear localization signal. And so working hypothesis was that if this is translated, it'll move from, there will be a start site and it'll move from exons 3 to exon 6, and it'll have the ubiquitin-like domain and the cut-like domain, but not the DNA binding domain. And so we asked the question, could this thing be translated? And so we work together with a core where the only way this thing would be translated, we put a tag, HYBID tag, very small tag at the end of this transcript. And the only way that it could be translated is if it starts from the start and moves all around and translates the HYBID. And lo and behold, HYBID is really nice because you could actually just treat your cells or lyse your cells and treat them with large bit. And you read out your fluorescent signal. And lo and behold, this is one of the days you're so excited in the lab because my student came in so excited and said she could read a signal. And so that's the only way, if this thing is translated, that HYBID would show up. And indeed, we're able to see that there was a signal from HYBID. We also have now shown with Western blot that indeed it's translated. It's a 22 kilodalton peptide. We also show that it's found in the nucleus. It has a nuclear localization signal. We also show that it's actively being translated because we see it associated with heavy polysomes. This is the circ3915 I just showed you. Its RNA, that RNA is associated with light and heavy. A circular RNA that is not translated is associated with just with the free, and SADB2 mRNA also with light and heavy, and TBP, tether binding protein, as a positive control is also associated with light and heavy, suggesting again and confirming to us that this circular RNA is translated. Because of time, I'm not showing you. We've also treated the cells with pyromycin that would dissociate your polysomes, and we actually see a shift from heavy towards free, suggesting that they're actually actively associated with polysomes. So again, just to summarize this part, what I've shown you is that this is what SADB2 would do. It could actually oligomerize with itself and bring several regions of the DNA close to the nuclear matrix. And by doing so, can actually bring an enhancer close to a promoter just by bringing several regions of the DNA close to each other. We also see that with AlphaFold that this peptide from the circular RNA can also oligomerize with SADB2. And if it does that, it might be acting as a dominant negative. In that case, what it would do is that it would oligomerize with SADB2, but what it would do is not bringing that, it will not bring DNA close to the nuclear matrix. And in doing so, might actually inhibit the effect of the function of SADB2 in the cell. So what I've shown you so far is that as we start treating cells with arsenic, we see that there's no circular RNA and there's no SADB2. But as we go through arsenic-mediated transformation, exposure and arsenic-mediated transformation, we see an increase in the circular RNA as well as linear transcript for SADB2. We think there's a circular RNA, microRNA, mRNA pathway to begin with. And we think possibly the circular RNA, when it initially comes up, it's possibly sponging a microRNA that otherwise would have targeted SADB2 for degradation. And we now have candidates that we are working to actually test this hypothesis. But what has actually always puzzled us is that when does it move, this circular RNA move from a non-coding function to a coding function? We think we got the idea last year where there was a paper published trying to understand myogenesis, and also SADB2 is important for myogenic differentiation. And what they saw was that during myogenesis, SADB2 transcript is highly made, the protein is the transcript is translated, the protein is made, but then the protein is degraded, and degradation of the protein actually drives this, acts as a signal to drive this differentiation process, and that's by caspases. We've now started data experiments in the lab where we're also seeing SADB2 being degraded, and we're also seeing caspases being activated. So could this be also a way by which the cell now tries to send the cells through to differentiation by arsenic? But we think, and then in that case, possibly the cell is thinking, I should have SADB2, but now I degrade SADB2, and the cell really wants to make SADB2. It now starts translating a circular RNA, and this circular RNA, the peptide generated from it, is very different from the full-length SADB2, and possibly inhibiting the function of SADB2. We think we're very grateful to NAEHS because none of this would have been possible without your support. We're actually discovering a lot of things through dysregulation by toxicology, but it's allowed us to discover novel chromatin regulators that otherwise would not have discovered if we were just using normal cell growth. I'm very grateful to the members of my lab. We're also trying to integrate all of this, what I just told you, to understand what exactly starts this process. We're now looking into metabolomics as well to see how we can integrate all of that to understand how arsenic is driving this process. What are the initiators, what are the drivers, and what are the things that maintain this process towards a carcinogenesis differentiation. And of course, none of this would have been possible without the lab, the support from Van Andel, my collaborators, Joop Dekker from UMass, Jonathan Lish for the histone proteins, Michael Puri for some of the Fred studies, the viral core for helping us. It wasn't trivial making those overexpression for the circular RNAs. Yaminini at National Cancer Institute for the AFM studies. And of course, again, very, very grateful to the NIEHS. With that, thank you so much.
Great. Thank you very much, Yvonne. That was really terrific. A lot of fascinating stuff. I think in the interest of time, we'll try to save questions for the discussion period at the end. And so we will turn now to our next speaker in this part of the symposium, which is Justin Colciano from University of Michigan. Justin is the John C. Searle Assistant Professor of Environmental Health Sciences, and he is an important member of and contributed to the Target 2 Consortium, and is also a former NIEHS ONES awardee. So Justin will tell us about epigenomics, single cell and single nuclear epigenomics in breast cancer health disparities. Justin, please.
Thank you so much, Trevor. And thanks to Fred for the invitation to present here. It's really great to see all this exciting science, and an honor to be a part of this distinguished group. And really thanks to AB and Fred for all their support. The work I'm talking about today is evolved out of my ONES grant, like Trevor was talking about, and I'm really thankful for NAEHS that makes this work possible. And then right from the top, I want to say that it's still like very, very early days in the use of single cell analyses in environmental health sciences. And so early to say that I would say the field of single cell epigenomics is very much a developing thing, just sort of set those expectations early. But today I'm going to talk about why the environment and breast cancer disparities is a topic. We think that single cell analyses may be particularly important analytical approach to consider. I'll give a brief overview about the use and challenges of single cell analyses in EHS, and I'll give a few vignettes of our current work in this space. And so a lot of our work here is really anchored in this knowledge that there are really substantial disparities in breast cancer survival. So these are data from the CDC's SEER that have been aggregated by Susan G. Komen, where just like looking at breast cancer survival by race, there are substantial disparities. If we, for instance, compare breast cancer mortality in Black women versus Asian Pacific Islander women here in the US, we see the Black women's risk of dying from breast cancer is about 2.5-fold higher. And folks have really dug in on trying to understand what might be the drivers of these breast cancer disparities. And one of the areas that folks have started to coalesce around is this concept of the subtypes of breast cancer, where we can take individual breast cancers, we can look at their gene expression profiles, and then group them into categories based on those profiles. And that's what's being depicted here in this early paper, but has now been sort of like validated across many, many more cancers, where what we see is that African-American women in the US are more likely to be diagnosed with a subtype of breast cancer called basal-like breast cancer. Oftentimes, this is also concordant with what we call triple-negative breast cancer, which is characterized by what it is not, unfortunately. So it does not express estrogen receptor, it does not express progesterone receptor, it does not express HER2, which takes away a lot of our toolkits and targets for treating this aggressive cancer. And so really trying to understand what might be the major drivers behind these aggressive breast cancers, these triple negative breast cancers, these basal-like breast cancers has been a major push in the field. And thinking then about the biological context, breast cancer itself seems to be a disease of dysregulated development. We can look at what we know about the signals that occur during early embryogenesis and embryonic development, right? We know that if we're going to move from a fertilized egg, a single cell, to a fully functional human with our trillions of cells, this needs to be a very tightly coordinated and regulated process, right? We have key signaling pathways that are taking us through this process that helping helping us regulate, patterning, angiogenesis and morphogenesis in this like spatiotemporal manner. And what we've started to see through all of this molecular profiling of cancer that's come out in the last 20 years or so is that cancer cells are using a lot of these same key regulatory pathways, but they're turned on at the wrong time, sort of like wrong place, wrong time for these regulatory pathways. What we started to learn through in-depth molecular profiling is that cancer cells themselves seem to exhibit behavior similar to stem cells during cancer initiation, progression, and metastasis. And then taking it back to that concept of aggressive breast cancers, like triple-negative breast cancers, these tend to exhibit more embryonic stem-like characteristics, or what we would consider cellular plasticity characteristics, where there's been experimental data that suggests that basal-like breast cancers actually come from dysregulated luminal cells that lose their identity and reacquire basal-like characteristics. And so this sort of like sets the stage then for trying to understand this. And so what we've been interested in is trying to get a handle on this at the normal mammary gland level. I'll just give you like a brief overview of the physiology of the normal human mammary gland here, because we'll be talking about some of these markers throughout the study. This is a picture adapted from Woodward et al. But really there's a couple major cell types in the breast. Here is what we would consider a terminal ductal lobular unit. These alveolar luminal cells here, this is where breast milk would be synthesized. Breast milk would then travel down these ducts lined by these ductal luminal cells. And then there's these myoepithelial basal cells that are compressing the duct and squeezing that breast milk to the nipple. I did my PhD research in Max Wish's lab, and Max's lab was the first to identify breast cancer stem cells. And his lab had done a lot of work on normal breast stem cells as well, and identified a small population of cells which express aldehyde dehydrogenases, which we call ALDH positive cells, as a potential stem cell in the breast, at least when we're looking at our functional assays. And so one of the early things that we were interested in doing was really try to characterize the heterogeneity within these ALDH positive cells. So we have a pretty robust FACS purification protocol. We would take dissociated reduction mammoplasties coming in at the hospital here at U of M, take those back to the lab, chop them up, dissociate them into single cells, and then run them through through FACS to purify out the cells with high aldehyde dehydrogenase activity. These are those cells that we think have stem-like characteristics. And then this was what I would consider like ye old days of single cell experiments, really one of like the first experiments to do this, where we leveraged this technique called the Fluidigm C1, one of the earliest single cell profiling methods, where you sort your cells into individual wells. You can visualize that you have single cells in these individual wells under the microscope. And then you can do, at this time, it was 96 gene qPCR for a targeted panel to really look at the expression of 96 genes across 96 cells. This was 10 years ago when we started this. This was like the very early days of single cell analyses in this space. And these were the data. These were like the big data at the time. I got these data back. We have 96 cells, 96 genes. I was a little bit overwhelmed. Remember, these were a sorted cell population out of the normal mammary gland. We weren't really expecting to see much going on, but when we clustered our cells in a hierarchical format based on their gene expression across these 96 genes, we saw that there was actually distinct expression signatures within the context of this one sorted cell population. This sort of like raised our eyebrows at the time, and we could like dig in a little bit more on this and start to see like, what are these different clusters? So we'd seen these like 4 different clusters. Cluster 1, I think these were cells where the amplification didn't work very well, but there were 3 other clusters that seemed to be quite robust. And so we could look and see one cluster, cluster 3, tended to express more of these mesenchymal like markers. So like Yvonne just told us about the EMT, the epithelial to mesenchymal transition. These cells tended to be more in that mesenchymal like state. We saw them expressing high levels of our stem cell markers like CD44, but then also high levels of mesenchymal markers like ZEB2 and SLUG and TWIST. We saw a population of more like epithelial-like cells, which expressed high levels of keratins and E-cadherin, MUC1, our epithelial transcription factor, GATA3. But then sort of surprisingly, we ended up identifying this cluster that we had to classify as hybrid because they were expressing sort of like all of our markers simultaneously. They expressed the highest levels of our stem cell-associated markers, like ALDH1A3 and integrin A6. They expressed high levels of our epithelial markers, like E-cadherin and EpCAM. They expressed high levels of our mesenchymal markers, high levels of luminal markers, high levels of basal markers, all of these simultaneously, along with some stemness factors like YAP1. And if you like ever think about like individual experiments that like push your career down a specific trajectory, this was one of those experiments for me, where we did this initial profiling. We had never looked at this before, and we ended up identifying that within this sorted cell population, there was a lot of heterogeneity, including cells that are floating around in the so-called hybrid state. And so we published this paper. We like worked on this a little bit more, and I was really excited when a couple of years ago, another paper came out from Joan Bruges' lab at Harvard, where they had really expanded on this. Right, they had done single cell profiling, but across all the genes in the genome, looking at single cells isolated from normal mammary glands from 52 individuals. And what they ended up doing when they did all of their unbiased clustering to identify all the different cell types in the mammary gland was that there were a couple populations in the mammary gland of what they call these basal luminal cells, these cells that express both basal markers and luminal markers at the same time. These are cells that we would definitely not ever pick up in bulk analyses, but there's a couple different populations of these. And then what they also saw that I think is quite interesting relative to environmental health sciences is that the abundance of these cells increases quite dramatically with age. And that's what's being depicted here on the plots on the left of this slide. We can look at the abundance of both of these basal luminal cell populations, see that they're tightly correlated with age. So like anytime we see those correlations with age in environmental health sciences, that sort of like raises red flags that these could potentially be environmentally modulatable. And so they then looked at the metabric, they looked at the metabric gene expression signatures, and what they were seeing was that these basal luminal cells, these cells that are enriched for both sort of like basal and luminal cell signatures at the same time, those are the ones that have the highest correlation with those basal-like breast cancers, those breast cancers where we see African-American women are disproportionately exposed to. So this gives us some hints that there may be some relationships here between the cell biology inherent in this sort of like condition where we've acquired both basal and luminal phenotypes at the same time in the same cell. These may be cells which have those same characteristics of those biological characteristics of those aggressive triple negative breast cancers. And so we can link this then back to our understanding of epigenomics. Right? We can put this in the context of the classic Waddington diagram, where we know in normal differentiation, we have our stem cell that lies at the top of the hill, right? The most potent. And then as that stem cell starts to differentiate, it starts to roll down the hill, it's acquiring characteristics. And then eventually our cells will settle in these valleys, right? These differentiated cells. In general, we think that this is a unidirectional process during development. But what we understand now in cancer is that this is not a unidirectional process, right? Oftentimes we'll see that cancer cells have de-differentiated. They've reacquired an embryonic stem cell like gene expression signature. This is the so-called cancer stem cell hypothesis. And then we also identify what we're seeing as transdifferentiation. So cells bouncing in between cellular states. So some sort of epigenetic plasticity within these cellular states that allows them to potentially move between states without having to revert all the way back to an embryonic stem cell state. And then what we might be seeing too is that cells are sort of like being able to balance on this hill, right, where they've actually acquired the characteristics of two or more cellular populations at the same time. So I think this really sets the stage then for single cell analyses to really start to dig in and understand what might be the biological factors which drive these. And we can think about that, of course, in the context of what we know about the investigated causes of breast cancer disparities. And so many people have looked at many different factors in breast cancer disparities. We know that breast cancer disparities are going to be complex and multifactorial. You know, Melissa Davis's group at Morehouse and others have done really impressive work looking at ancestry-associated genetics, especially West African ancestry-associated genetics, and looking at the risk of aggressive breast cancers related to the composition of the tumor microenvironment. Others have looked at the impacts of changing obesity trends, access to healthy food, changing reproductive trends, where we know that all of these factors are potentially associated with the risk of developing those basal-like or triple-negative breast cancers. Of course, we know that access to care, early screening, detection, and treatment are also all really important, especially when we're thinking about disparities in breast cancer mortality. And folks are starting to explore the impacts of systemic racism, social inequality, and the social determinants of health on breast cancer disparities, because we know that the stress-related pathways that are dysregulated from sort of like combined social stress, also are a lot of the same pathways that can promote breast tumor growth. What's a little bit less understood and kind of like where our lab has been working in this space is under the area of environmental exposures. And of course, we know that chemical exposures are not distributed equally. Sort of like where you work, where you live, these are things that really drive your risk of cancer, right? This is a picture of Marathon Oil here in Southeast Michigan with a playground right in front of it. Similarly, Cesar Chavez High School in Houston down at the bottom right. You know, Rena Jones, our collaborator at NCI, just published a really important paper in JNCI showing that there's disproportionate exposure in proximity to hazardous waste sites in minoritized populations. So obviously we know that a spatial localization like we heard about from Shanshan earlier really drives an individual's risk of cancer. We can think about the water we drink, right? This is the pictures of contaminated water in Flint, you know, an hour up the road here from Ann Arbor, where we know that, you know, the population of Flint was disproportionately exposed to lead. Yvonne just told us about arsenic contamination in Michigan in the groundwater, and then the food we eat, and that, but also a lot of the products that we're using on a daily basis. All of these things can impact our risk of disease. We know that these exposures are not distributed equally, and then we can host this, of course, in the framework of all of the potential chemicals that we might be exposed to. Sort of like thinking within the context of these like unbiased expose-on-wide approaches, can we use these to really characterize these unequal distributions of these chemical exposure disparities? We've tackled this a little bit as part of my ONES grant. So an outstanding bioinformaticist in the lab, V. Wynn, who's currently doing her postdoc in Chirag Patel's lab at Harvard, went and downloaded all of the CDC's data on chemical biomarkers in the US population. We ended up having data on about 38,000 women across 150 different chemical biomarkers across all of these chemical classes. At the top, things like brominated flame retardants, persistent organic pollutants, metals, phthalates, things that are in personal care products, VOCs, PFAS chemicals, sort of like you name it. And then V wanted to do like comparisons between demographics, right? And see like, are there areas where you can pick up profound exposure disparities? And certainly one of the most profound that we saw was specifically in non-Hispanic Black women in the US. And that's what's being depicted here on this volcano plot. So chemicals that tend to have higher levels in non-Hispanic white women come out on the left of this plot. Chemicals that tend to have higher levels in non-Hispanic Black women come out on the right of this plot. And there's about 50 different chemicals across multiple chemical classes. Some of these are known carcinogens. Some of these haven't been classified at all for carcinogenicity, but we see levels of some of these chemicals that are 350% higher on average in the US population. In non-Hispanic Black women. So this gives us an idea that this is probably an area that we should be exploring with respect to breast cancer disparities. So I'm going to tell you a little bit about how we can take these data to inform our toxicology experiments, to inform our environmental health studies, using single cell and tissue spatial methods to investigate environmental exposure disparities and this idea of cellular plasticity or reacquired stemness at the single cell level. And so I'll just give a quick overview about like what is out there for us to be using when we're considering doing one of these studies. So I'll tell you a little about a few different single cell methods. So there's single cell methods that fall under the universe of what we would consider well-based methods. So the Fluidigm C1 data that I showed you, so like at the beginning of the presentation where we used microfluidics to sort cells into individual wells, that's like the classic well-based method. There's other methods like this called the isolate that allows you to do very in-depth profiling of a few individual cells. Others have gotten pretty creative about how they do this. So Parse is a biotech company that does what they call combinatorial indexing or split pool barcoding, which is a technique that enables us to start to analyze hundreds of thousands or millions of individual cells, like a very exciting technique. Many folks in the field have been moving towards droplet-based methods for single cell or single nucleus profiling. This sort of like originated with DropSeq out of the McCosker lab at the Broad Institute, but more recently, sort of like the commercial version of this that everybody's been using is from the 10x Genomics Chromium device. And so I'll talk a little bit about these data. But now there's this huge explosion of this new type of data, these like spatial data. And so these aren't the geographic spatial methods. methods that Shanshan was telling us about, these are cellular or tissue level spatial data, where we can get very in-depth information using imaging-based approaches or sequencing-based approaches to really dig in on the spatial distribution of markers, RNA, protein at the single cell level. And so some of these, we could group them under what we call morphomics type of measurements. These could be grouped under the area of high content imaging. I think like cell painting falls under here. There's multiple antibody-based approaches that allow us to measure multiple proteins. So the Polaris gets us up to 7 or 8 proteins at a time. PhenoCycler gets us up to 50 or 60 proteins at a time. Hyperion is similar there. So there's ways to do sort of like high resolution, sort of like multi-layer mapping of protein expression. And then there's other new and emerging sequencing-based methods that really start to allow us to dig in a spatial way on RNA expression. The classic examples here would be like the Visium from 10x Geomics. SeqScope is a method that was developed here at U of M in-house, and DBitSeq that came out of Ron Fan's lab at Yale. These are very powerful techniques to dig on this, and then a whole universe of new in situ hybridization-based approaches that can give you up to single molecule resolution of your markers of interest across a tissue section. So a lot of like very exciting approaches here to dig in on, but I think the reason that these haven't been sort of like uptaken as much in the field of EHS is because I think we have specific challenges within the context of EHS that makes using these single cell and spatial approaches you need to like at least give it a little bit more thought, right? So in contrast to what we've been seeing with these like incredible atlasing projects like the Human Cell Atlas, where we're defining all the cell types in the brain or the heart or the mammary gland, when we're doing an environmental health study, oftentimes we're dealing with more modest effect sizes of transcriptional changes, right? We're comparing between an exposed and unexposed condition. Those are not going to be the same sort of transcriptional changes that we might see in between cell types, for instance. So this is something that's a challenge. Oftentimes, these single cell methods have a limited ability to detect lowly expressed genes. We know the biological replicates themselves can be costly. So if we're trying to add in multiple biological replicates, that can make it challenging. We know that the sample handling itself can be challenging. How you do the tissue dissociation matters. We know that batch effects can compound data analysis. So thinking about how do you generate all of this data in a way that you can get useful information out. And then really the data analyses themselves are complex and rapidly evolving. And so the way that we've been doing it from the toxicology standpoint is really thinking about, can we build a living biobank of normal breast samples, partnering with the Komen Normal Tissue Bank to allow us to assess toxicant impacts on breast cancer disparities. And so I just want to take a second to hype up Susan G. Komen. One of my mentors, Michelle Cote, recently took this over. And this is like an incredible resource for folks that are doing breast cancer work. So they have normal breast biopsy samples from volunteers from thousands of women, paired with nice epidemiological data relevant to what we were doing. Some of these breast tissue biopsies have been cryopreserved, so you can like grow them up in culture later. And that's what we were doing here was growing up very tiny breast biopsies from African-American and European-American women who were premenopausal and nulliparous. We wanted to keep everything sort of like as similar as possible as we could. We grew these up via conditional reprogramming, and then we profiled them for ancestry informative markers. Komen did that. We did single cell RNA sequencing all of these, profiled them for genetic polymorphism, so the global diversity 8 SNP array, which we can then impute their genotypes via TopMed. Then we did DNA methylation for epic arrays. So we have like baseline data on everybody. And so if you're interested in sort of like the application of these types of approaches, I would highly recommend checking out the Susan G. Komen Tissue Bank, which is an incredible resource. And so I'm going to tell you a little bit about how we've been using these in the morphometric context, really trying to think about, can we use like imaging-based approaches at the single cell level to get a handle on this idea of cellular plasticity or sort of like dual cellular identities. And so for those of you that aren't as familiar, morphometrics or high content imaging is a very promising tool that I think like has a really important place in EHS, but hasn't been widely applied yet. And so here, how these generally work is we're going to be looking across genetic, perturbations, or more likely in our case, of course, chemical perturbations, being able to run these experiments in high density. So like we'll oftentimes be running these experiments in a 384 well plate. We'll do high content imaging. We'll then use computer vision type of approaches for our image analysis, which can then give us very in-depth morphological profiles on many cells. So we just like wrapped up one of these experiments and got data on over 1 million cells, for instance. So like the scale of analyses that we can do with these high content imaging approaches are very good, very high scale, and also gives you a really good opportunity to detect rare phenotypes in your data if you're thinking about dealing with rare cell populations. The things that we can look at include cell structures. We can think about things like RNA, the endoplasmic reticulum. This is sort of like the classic cell painting. On the left, we can look at cell state markers, like our markers of luminal and myoepithelial cells. I'll tell you about this like keratin 8, keratin 14 panel we've got. We also have a similar panel working with stem cell markers that I'd be happy to talk about later. And then you can, like, if you have a hypothesis, you can add on different type of epigenetic readouts like nuclear morphology, nucleolar morphology. You can look at different histone modifications. You can add in different transcription factors. And so just to give you an idea about how one of these experiments could work, this was work that was done by Jade Schroeder as part of her master's thesis, and Annika Topaswi, who's a research scientist in the lab. And they optimized this method where we could just do a simple stain with stain for DNA for cell segmentation, stain for keratin 8, that luminal cell marker, and a stain for keratin 14, that myoepithelial cell marker. We can then look at these guys simultaneously and then use a random forest classifier to identify, hey, within our sample, how many cells are falling into these different groups. These like luminal cells that are expressing keratin 8, these basal cells that are expressing keratin 14, and these cells that seem to be in this hybrid state, expressing both keratin 8 and 14 at the same time. And so just to like highlight why this might be important for those of you who are in this, we can look at the expression of these markers across different samples, right? So this is one of our samples from our African-American study participants that's predominantly luminal. Here's a sample from our European-American participant that's predominantly myoepithelial. And then we have samples that sort of like float in the middle, right? Primary cultures themselves are heterogeneous. We even see that cell lines are heterogeneous too. So that we think that this is a really important thing to take into account when we're doing this type of work. You know, here we would come in, we do toxicant exposure coupled with high content imaging. So just to give you an idea about how accessible these methods are for Jade's thesis, she did 6 different lines, 8 chemicals across 4 doses and 3 technical replicates for a 48-hour treatment. And then we can start to see how do chemical exposures where we see disproportionate exposures in Black women. So one of our chemicals of interest here was bisphenol S. How does that perturb sort of like these hybrid states? And so what we see here, for instance, is in this participant, this African-American participant, we see sort of like this dose-dependent increase in the hybrid population. So it really allows us to test for alterations in cellular phenotype and chemical by individual, really at the single cell level, where we can then think about layering in extra measures on the epigenetic state. And so I want to just touch briefly on using droplet-based methods as like an orthogonal tool to start to dig in on this. And there are some nice tricks we can do in the context of these droplet-based methods that can give us some ideas here too. And one of them is this concept called genetic multiplexing, which allows us to really scale up the number of biological replicates that we're running in one of our experiments if our samples are coming from genetically distinct individuals. So it's like perfect for these types of experiments where we're dealing with this like living biobank of human samples. You can imagine something similar with iPSC derived lines, but here we're taking individual cells from genetically distinct individuals, we're pooling them together for processing. As we sequence them, we can then call the genetic polymorphisms from the sequencing data back, and then take those polymorphisms and link them back to the data that we already have from each individual, right? We already have everybody's genetic sequence. We can tell which cell came from which person. So that really gives us some power to identify doublets in our sequencing data, because these cells then have combined genotypes because we have 2 cells from different people in the same droplet. And there's multiple bioinformatics tools for this purpose, like DMUX was developed in-house here at U of M. And so Tasha Tong, who was a former PhD student in the lab, was really interested in taking this approach to really understand the toxicity of different bisphenols, BPA, BPF, and BPS, like I was telling you about, on these normal mammary cells. She came up with this like wild experimental design where she wanted to treat cells from 6 different people by those 3 chemicals, by 3 doses plus control. So like 60 conditions of single cell. So if we were going to run 60 individual single cell experiments, that prep alone would have cost over $120,000. Tasha figured out a way we could get this all in one single tube. So like enabling sort of like this high throughput type of approach that I think we need, that we need in EHS. So we culture our samples from our 6 individuals. We can pool them. We then dose them in 12 different flasks with sort of like 4 different concentrations of these 3 different chemicals. We can then use some reagents from 10X that they call CellPlex, where we have oligo tagged lipids, and each sample gets its own little oligo tag. So then we can then differentiate which samples came from which flask later. We then pool everybody together. So now we have like our 6 individuals across our 12 different treatment conditions, do our single cell encapsulation, library prep, and sequence, and then can deconvolve. So this is like pretty incredible that we can now do this like sample multiplexing in the context of a single cell experiment, because it really helps us do an individual plus cell type specific concentration response evaluation. And so we can then look at our single cell data, these UMAP plots. Each dot is an individual cell. We can look for our key marker expression. So here we can see that keratin 5 tends to pop up on the western edge of this UMAP. Keratin 8, our luminal marker, tends to pop up on the eastern edge. We can start to see, hey, are there cells that express these hybrid populations here being depicted in the green? And then just like we had seen in the Abuja paper, we can see that those cells that are sort of like hybrid for keratin 8 and keratin 5, those are the ones with the highest signature for that basal breast cancer signature, right? Giving us, again, this idea that there's something going on with those cells. We can then look and see, like, is it possible that the environment might modulate this, right? And so we can do single cell gene set enrichment. We can look for example, enrichment across embryonic stem cell genes. That's this long embryonic stem cell core, or we can look for enrichment across these basal breast cancers. And indeed, we do see that low dose BPS, for instance, can enrich for an embryonic stem cell like gene expression signature or a basal like gene expression signature. So that like really lays some groundwork for us to then start to follow this up with orthogonal experiments and methodological approaches to really dig in on this. And the one that we're really excited about single cell multiomics. And so multiomics means that we can now get multiple layers of omics from an individual cell. And in this case, it means that we can get simultaneous profiling of mRNA and chromatin accessibility, like ATAC-seq data from the same nucleus, which is pretty incredible. And so this is a method from 10X where we're now able to sort of like simultaneously generate both of these data types, but it gives us like a handle on some really fundamental questions, I think, that we're going to need to understand. So we can start to understand like what epigenetic programs are driving these hybrid states, right? We can think about which transcription factors might be activated in these cellular plasticity programs, like how is this basal luminal cell existing? We can think about, do environmentally induced hybrid or stemness programs mimic basal breast cancer epigenomes? We can think about, are the epigenetic programs that are activated in these hybrid cells the same as those that are involved in epigenetic aging, right? Do we see this increase in those cells with age because it's those same sites that we know are associated with epigenetic aging? And then we can really think about, on a tissue level, how do these signals from the microenvironment promote and stabilize these hybrid cells? And I think this is what's taking us sort of like to this next frontier of single cell and spatial multi-omics, right? We can then talk a little bit about, just briefly, these sequencing-based approaches, because I think these are going to have high applicability in environmental health sciences and toxicology. And so for those that aren't familiar, quick overview, these single cell based approaches, right? So the 10X Visium is one. Usually we're going to be dealing with some sort of like barcoded spot approach. So we're going to have a capture area. We're going to lay our tissue on top of those barcoded spots. Each spot has its own oligo. So then any RNA that incorporates that oligo, we sort of like know where it came from on the spot. So we have like the XY location of where that tissue was and where that RNA came from. So it gives us really nice data. So like whole transcriptome data on about 5,000 of these spots. Each spot in the context of the Visium is about 55 microns in size. So, somewhere around 10 to 15 cells. We have a capture area of about 6.5 millimeters. The data are discontinuous. You can see those spots are like spread apart, but there's bioinformatic approaches for imputation. I'll show you on the next slide. But the advantage here is you have the potential to probe additional omics layers beyond RNA. So we're working with Atlas Exomics, which is a company that's spun out of Rong Fan's lab for the DBIT-seq platform, and we're running spatial ATAC-seq, for instance. Those data are a little bit undercooked to show here, but I'm saying that it's possible to start to generate these type of data. But the data that you can get from here are incredible, right? So this is like whole transcriptome. We're just showing a subset of these data here. And so we can work with, we have an awesome collaborator in biostats here, Shang Zhao. Shang has a NIGMS R01 that I'm a co-I on that's really designed to develop new methods for spatial omics bioinformatics analyses. And so he's developed a technique called SPARC, which allows us to identify genes that are expressed in a spatial manner, and a technique called CARD that allows us to do spatial cell type deconvolution and gene expression imputation or smoothing across the tissue, sort of like analogous to the Krieger example that Shanshan was telling us about. So David Aguilar, who's a PhD student in NAEHS, and Peiyao Zhao, who's a PhD student in biostats, have been working on this. And these data are super rich. And just to give you an example, we can look at our classic cell type markers, but we can look at all these different markers of environmental stress, right, and see how they spatially localize across a tissue. We can look at inflammatory markers. We can look at our stemness markers. And now we can start to understand really how do the signals from the microenvironment help to promote these states, and are these states sort of like potentially modulatable by environmental factors. So we have like a series of like 10 triple negative breast cancers currently at the core that we're going to be able to like now integrate these data in with. We're quite excited about this approach. And so this is sort of like a busy figure, but just to highlight kind of like where we think that this is going. If you're interested, so Patrick Allard, Christy Willett, and I are co-editing an edition of Current Opinions on Toxicology and Single Cells in Toxicology, which is coming online now. And David made this figure, but sort of like conceptualizing how this can work, right? We look at sort of like exposures across all of what we understand about exposure disparities. We can look in animal models, in vivo models. We can look in human mammary tissue, and we can think about this sort of like across the life course, digging in on the tumor microenvironment. We can dig in there, really do the single cell transcriptomics, which helps us do cell state identification. And we can do the cool experiments like looking at changes in pseudotype, right? Looking at differences in cellular trajectory and stemness state. From these tissues, though, we can also do that spatial transcriptomics, which gives us sort of like the spatial view of the total expression profiles. We can then, oops, sorry about that. We can look at the potential for ligand receptor interactions to see like, are there cells within a little microenvironment that might be promoting each other through ligand receptor interactions. We can look across different neighborhood motifs to see which neighborhoods are enriched for these these like plastic or stem-like states. That really gives us this new method to evaluate environmental drivers of aggressive breast cancer. And so just to highlight, if you're interested in, we have a program here at U of M that's open to anybody, the Single Cell Spatial Analysis program. Check us out at singlecellspatialanalysis.umich.edu. We're trying to build a community now, more than 200 researchers strong, of folks that are interested in applying single cell and spatial approaches in their work. And with that, I'd like to thank everyone in the lab that does it, our collaborators on the ONES grant and the renewal, especially NIEHS, but other NIHs for funding our work. And if there's time, I would be happy to take any questions.
Thank you very much, Justin. I think we're actually running a little bit behind. So if there are questions, we'll ask you to save them for the council discussion. So we'll go with our last presentation this afternoon now, which is Victor Corzes from Emory University. Victor is professor in Department of Human Genetics and has really been a pioneer in helping us to understand about transgenerational inheritance, along with some really sort of fundamental work that his lab has done. So without further ado, Victor, the floor is yours.
Sorry, I couldn't unmute.
We've lost your slides, Victor.
Yeah. Okay, can you see the slides now?
Yes, you need to go into presentation.
Yeah, thank you, Trevor. Okay, so I don't have to convince this audience. Can you hear me okay? Yeah?
Yes, we can.
Yeah.
That's good.
That the environment is important for disease. But most of the time we tend to think about the effect of environmental exposures on the person who's exposed, and maybe they are exposed in utero to the newborn. And what I would like to talk about today is transgenerational effects. That is to say that inherited disease state may have a non-genetic origin and is caused by environmental effects that cause germline epimutations. And there are a lot of examples, both in laboratory animals and also in humans. For example, I have down here the over-calyx studies that show this, but there are not very many convincing mechanisms for why this can happen. So one example of this is autism. So if you look at autism, this is an example in California of people that are in the DDS services department. So these are people that are in California who are not able to live on their own, and they are held by the state. And they have the state of California has a very strict requirement for who can be part of the DDS services. And so these are all phenotypes classical of severe autism. So the numbers that you see increasing here from 1987, very few cases, to 2020, where there are almost 140,000 cases. They are not caused by differences in the diagnosis of the disease. So if we look at genetics, and people have done GWAS studies with autism, only around 18 to 20% of cases have a mutation in any one of the now almost 1,000 genes that have been identified as being responsible for autism in humans. So the other 80% are unexplained. And it is possible that the origin of them is epigenetic in nature. So to study this, we are looking at obesity in the case that I'm going to show you today. We are looking at obesity, not autism. So we follow a paradigm that was first published by Mike Skinner, and now has been studied by a lot of different people, including Marisa Bartolome, for example, who did a beautiful, very careful analysis of this. So if we expose pregnant females at a time that I'm going to show you in a minute, when the embryo is going through reprogramming of the germline, we expose them in this case to BPA. The F1 generation, which Mike Skinner and others have shown that they have an increase in breast cancer and testicular cancer and various other things, it's not obese. But if you now cross this mice, the F2 generation is obese, and I'll show you more detail on this, and the F3 generation is obese. And we can transmit this presumably epigenetic change that has happened in the germline that causes an obese phenotype. We can transmit it through both the male germline, if we take an exposed male and cross it to a non-exposed female, the next generation, this is an F1, the next generation is obese, and the other way around. So the change in the epigenome of the germline has occurred both in the male and the female germline. So I'll go quickly over this because, as I said, has been published, and I'll just make a few important points for my talk. So this is the visceral fat tissue in a control animal. This is F4 generation male, and you can see the difference in visceral fat tissue. They have more adipocytes, and the adipocytes are bigger, so they have higher lipid content. And you can see here the difference in weight. Again, these are F4, and the difference in visceral fat. And the red in my diagrams is always the median. So, as I said, a few points for things that are going to be important in the discussion. So we've done this 6 times, and with similar results in all 6 times. And the different times that we've done this are in different colors. The red again is the median. You can see that control mice, these are always males, because the effect is not very pronounced in females. Have a range of weights. In the F1 generation, the range is more or less the same. In the F2 generation, the F3, the F4, what you see is that the range increases and the median weight increases, but in each generation, there are few mice that have the same weight as control and in the same range as control, a few mice that have very high weight. If we cross these mice that have very high weight, we get the same range. We get, from those mice, we get mice that have a weight similar to control and also very high. Okay, so we are not selecting for some genetic variant in the population. If we cross this mice that have low weight, is the same. Okay, now the median weight is highest in the F4 generation, and then as we move to the F5 goes down, the F6 is go down, the F7 is this has the same range of weights as the wild type with the same median weight. So the F6 generation, the sperm presumably doesn't have the epigenetic alteration, since everybody in the F7 generation now has normal weight. And if we look to see at the change in weight with age, and this again are F4 males, you can see that at 3 weeks of age, when we wean them, the weight of the controls and the BPAs, which are in blue, are more or less the same. And as they age, the difference in weight increases. Okay, so you can see the BPA mice now have a much higher weight than the control as they age. So this suggested to us that maybe the problem was an issue with food intake. And when we measure food intake, you can see the control, the difference between control and F4. The F4 ancestrally exposed mice to BPA have a much higher food intake than the control. And that food intake seems to happen at a specific time during the day life cycle, so that the mice, instead of going to sleep in the morning, the BPA mice stay up munching and they eat more than the controls. There seems to be a difference in the dark, but it's not statistically significant. So presumably what is happening, what BPA is doing to these mice is that it's affecting something that is controlling appetite. Now, if we think about the epigenetic, what is responsible for the epigenetic memory of obesity between these different generations? The most obvious explanation is DNA methylation. So I have a bias about this. Although RNAi has been it has been characterized as being responsible for transgenerational transmission of phenotypes in C. elegans, the sperm only gives 5 to 10 femtograms of RNA to the oocyte, to the fertilized oocyte, while the oocyte has around 1 nanogram of RNA. So the amount of RNA that goes from the sperm to the fertilized oocyte is very small compared to what is in the the oocyte. Now, and histone modifications, the way we see them, most of them, especially the active modifications, are a consequence of transcription rather than the other way around. So DNA methylation from, and this is our subjective view of this, is the most likely memory carrier of epigenetic memory. Now, there is a problem with DNA methylation because right after fertilization, so here on the y-axis, I have DNA methylation. Right after fertilization, most CPGs in the genome, in the paternal chromosome, they are actively demethylated, and in the maternal chromosome, they are passively demethylated, such that most methylated CPGs, with the exception of imprinted CPGs, by the implantation stage, by the epiblast stage, are erased. And then they are reestablished again, not in the same places as before, by the epiblast stage. Now, those are then more or less stable in somatic cells, but in the cells that are going to form the germline, in the PGCs, they are demethylated quite rapidly, and even the imprints are demethylated. Now, the time when we expose the mice is this box here between day 7.5 and 13.5. And so we reason that BPA, which is, interacts with the estrogen receptor and the glucocorticoid receptor, and it antagonizes the androgen receptor, is causing a transcriptional change in the PGCs directly or indirectly at this time. And that will cause new transcription factors or transcription factors to bind to new places in the DNA. Now, at this time, the DNA is demethylated, and the transcription factors will be able to bind freely to any site in the genome if it's a transcription factor that recognizes a motif that has CPGs and it interacts with DNA in a CPG-dependent manner. Now, when this site where the transcription factor is bound is remethylated, the presence of the transcription factor will not allow that site to be remethylated, and of course that will depend on the affinity of the transcription factor and on the occupancy, of the transcription factor, the KD for binding to the DNA, and so that site that would normally be methylated now is not methylated and is demethylated. So what constitutes the memory, in spite of all this reprogramming, is an interplay between DNA methylation and transcription factor binding, so that the transcription factor can keep the memory at times when the DNA methylation is erased. So a number of years ago, we showed that sperm RNA it's known that a few nucleosomes remain, and we and others show that these nucleosomes are located in special places. They are flanking promoters, and they are flanking transcription factor binding sites and enhancers, and they have histone modifications, and there are transcription factors present, and CTCF is present there, and cohesin is present there. Now, how do we figure out if there's a transcription factor or more that remain bound to the sperm and the oocyte after BPA exposure? So what we decided to do was to do a ATAC-seq in a sperm from control F1, F2 through F6, and also in an IVF experiment that we had done that I didn't show you, and look at genome-wide all the ATAC-seq sites, so that ATAC-seq gives you, tells you the presence of transcription factors at chromatin-accessible regions. And when we analyze all that data together, we found 12 different sites that are different between BPA and control. And then when we did the same thing in the oocyte, only one of those was maintained, was present both in the sperm and oocyte. And that is the CTCF site right here that is present in the gene FTO, which you guys heard about before. So FTO is a gene that is associated with obesity in humans. There are variants in humans in the FTO gene, in the first intron of the FTO gene, that are related to obesity. They are next to a silencer element. In our case, the changes are in the 8th intron, and they are associated with an enhancer. That's why some people say, well, FTO does the opposite thing in humans and in mice. Is not really true. You just have to understand why it's doing different things. Anyway, this CTCF site, you can see, is not present in control and is present in all these obese animals. And then the F6 generation, it goes away. And if we get fat animals from an IVF experiment in which we took a sperm from BPA, treated and controlled oocytes, and you can see that is also here. And if we do ChIP-seq, you can see that, so BPA, ATAC-seq only allows us to find a motif for CTCF at the summit of the peak of the ATAC-seq peak. But if we can confirm this by ChIP-seq, this is BPA from an F3 male, and you can see the difference between control and BPA. Now, if you look to the right of this CTCF site, there are all these different ATAC-seq sites that, you know, they are present in control, but they are really induced by BPA. All these other sites correspond to binding sites for estrogen receptor, PPA gamma, androgen receptor. Okay, so things that make sense in the context of a BPA exposure. So we think that this is an enhancer that is right next to the CTCF site. Now, if we look at DNA methylation right at this CTCF site, the CTCF site has a CPG in a subset of sites in the genome, and that CPG is, when it's methylated, it completely inhibits DNA binding by CTCF. You can see this site, this specific CPG here, is mostly methylated in control, is demethylated in BPA3, and it becomes methylated again in F6. So this makes sense in the context of the hypothesis that I told you. So the idea then, the idea to explain the transgenerational inheritance is that BPA exposure causes a transcriptional change, causes the activation of new enhancers. And in unrelated work, we've shown that during cell differentiation, when you activate a new enhancer, you activate, demethylate a binding site for CTCF, and then CTCF binds so that it can bring that enhancer to a target promoter. Now, for our hypothesis to be right, it would have to be that this CTCF, the occupancy by this CTCF site, is transmitted from one generation to the next. Okay, it will have to be transmitted so that it stays bound in the germline, and also it will have to be transmitted to the somatic cells. It will have to stay in the somatic cells that are the target tissue for the phenotype that we are looking at. So CTCF, as Yvonne mentioned, makes loops to organize the chromatin and to bring enhancers to the target promoters. If we do high C in BPA and in control, and we do a subtraction map to look at interactions that are present in BPA but absent in the control, we can see that this CTCF site, this is the FTO gene, now contacts a series of genes on this other side and its own promoter. And among the genes that it contacts is a gene called IRX3 and IRX5. Now, I'll come back to this later, but the reason why we thought that that was interesting is because IRX3 and IRX5 are involved in the differentiation of appetite controlling neurons. So you can see here, this is a qualitative analysis, this is a more quantitative analysis. looking at interaction differences from the CTCF side between BPA and control. Okay, there is another important component of this that we realize eventually, and that is that this is, again, the FTO gene IRX3 and IRX5. There is another enhancer here that we call the distal enhancer, and this shows you ataxic changes in this distal enhancer, and you can see a slight increase between control and all the other obese generations, and a decrease again in the F6. Okay, so, and these binding sites, these different ataxic peaks have binding sites for androgen receptor C and CTCF. And from the High-C data, we can see that this distal enhancer interacts with IRX5 in a BPA-dependent manner, interacts with the other enhancer inside the intron, and it interacts with the FTO promoter. So it has a possibility of regulating FTO and IRX3 and IRX5. Now, what is interesting about this enhancer is that it makes an enhancer RNA. But the other important thing is that FTO is an M6A RNA demethylase. And so in a sperm, FTO is upregulated due to these loops. And when we look at RNA from sperm, we see a dramatic decrease in M6 RNA methylation decrease. This is, look at the scale here. This is log 2, 2.5 and 5. And most of those RNAs are non-coding RNAs. That is in this slide here. I'm sorry, I'm going a little fast, but I want to keep within my time. So one of those non-coding RNAs is an enhancer RNA. And that enhancer RNA is in this FTO distal enhancer. So you can see this is the enhancer RNA within this enhancer. So this enhancer RNA is the same in BPA and control, but the M6A methylation state is dramatically decreased in mice that have been ancestrally exposed to BPA. So BPA is affecting the methylation state of this RNA. And another thing that is important is that this RNA is chromatin bound. So you can use 4 molar urea, and this RNA is still stuck in the chromatin. So that when it's transmitted to the sperm, to the oocyte, the RNA goes with the sperm chromatin to the oocyte. It doesn't go as soluble RNA. Again, this RNA is in this distal enhancer. Now, this is interesting because Chuan He's lab published this paper where he showed that when the enhancer RNAs are demethylated, they affect the accessibility of the chromatin, and that these RNAs, the M6A methylation of these RNAs can affect chromatin state and enhancer function and transcription. And so now the problem that we have in front of us is complicated by the fact that originally we had a simple hypothesis. The hypothesis was that the CTCF site would persist from one generation to the other. Now, it is very likely, and we have mice now that we are analyzing in which we deleted this enhancer RNA. It's very likely that the M6A modification of this enhancer RNA is affecting its interaction with FTO and is affecting the inheritance. Now, if all this is true, if we delete the CTCF site, you expect the whole thing to reverse. So we made mice that have a 300 base pair deletion, in which we deleted the CTCF site and a FOXA1 site that is next to CTCF. And in a different set of experiments, we have shown that recruitment of CTCF site to a new site in the genome, recruitment of CTCF to a new site in the genome that is methylated requires pioneer factors like FOXA1. So it probably FOXA1 also has an important role. Now, these mice in which we deleted those two sites, now when we expose them to BPA, they don't gain weight. Here's an experiment, the blue and the orange, whereas the control mice, the wild type mice, gain weight. And you can see if you delete the CTCF site, the visceral fat is the same when you expose them to BPA. The adipocytes are the same. And now they don't gain weight as they age. So the FTO deleted lines are the blue and the orange, and you can see they are undistinguishable, whereas the wild type BPA, which is the green, gains weight. And if we look to see if they eat more, you can see that now these mice that have been exposed to BPA, but they have a deletion of the CTCF site in FTO, don't eat more. They eat the same as the wild type. So we have been able to revert the physiological response to BPA by just deleting those 300 base pairs. Now, of interest is the fact that the deletion of FTO of the CTCF site now, of course, does reverse also the molecular phenotype on N6A methylation. So you can see now this RNA in the BPA mice with the FTO CTCF deletion doesn't get demethylated N6A, and in this enhancer, the ataxi signal doesn't increase anymore compared to wild type. Okay, so let me summarize this with a diagram. So we have FTO, the FTO gene, and IRX3 and IRX5, and here is the enhancer. We don't know exactly all the molecular events that have to take place, but BPA binds maybe to the estrogen receptor or the glucocorticoid receptor. It recruits FOXA1, then that recruits CTCF, and now CTCF can bring the enhancer that is next to it to IRX3 and IRX5 and to its own promoter, cause their overactivation. Now the FTO protein is translated and is able to demethylate the enhancer RNA plus other RNAs in the genome. And now this enhancer RNA with N6A demethylated opens the chromatin and is able to recruit additional transcription factors that interact back and form a positive feedback loop that is probably responsible for the transgenerational inheritance. Now, I'm not keeping track of time. I forgot when I started talking, but let me just take 5 more minutes.
You're very close, Victor.
Okay, 1 minute. So how about the target tissue? The target tissue is the hypothalamus. That's where you, that's where the neurons that control appetite, which are these POMC and AGRP NPY neurons, which are here in blue and purple, and they differentiate from these other cells that are called tanocytes and radial glial-like neural stem cells. So we did multi-omic analysis, single-cell ATAC-seq and RNA-seq, and found a lot of changes that I won't go into, But an interesting change is the fact that there are many more AGRP neurons, which is something that one should not conclude just from single cell data, but we confirm this by immunofluorescence with AGRP antibodies. So this is wild type control. You expose to BPA more neurons, you delete the CTCF side back to control, you expose them to BPA back to control. Okay, now these neurons, for their differentiation, people have shown that they require oligodendrocytes in the hypothalamus. So you can see in these oligodendrocytes, this is the CTCF side. So this is an ataxic from the multi-ohm data. Control, and this is the CTCF side that we saw in the sperm. And this shows you that it is possible that the CTCF side perdures, stays bound in the somatic tissues. And we haven't looked at DNA methylation yet. Now, interestingly, in these precursor cells, in these radial glial cells, the CTCF side, these are the ones that give rise to the appetite controlling neuron. The CTCF side is the same in control and BPA, but a very another CTCF site that is in close proximity is induced by BPA. And what is interesting is if you look at mice, the mice that in which we deleted the CTCF site, this one is not induced. So there are these two correlations between the phenotype that we see, the increase in AGRP neurons and the persistence of the CTCF site in oligodendrocyte and an adjacent CTCF site in the radial glial cells. So I'm sorry if I went over I finished. Let me just emphasize the fact that the problem is really complex, and it's probably a combination of DNA methylation, transcription factor binding sites, and epitranscriptomics that is able to establish this feedback loop so that the epiphenotype induced by BPA is transmitted from one generation to the other. And let me thank Yoonhee Jang, who did most of this work, and Xiaoling Wang and Daniel Ruiz, who also contributed to part of the work. And of course, NAEHS is for funding this specific grant on top, specifically funds this work. So thank you.
Victor, thank you very much. It's really fascinating work, and I think it'll serve a good way to take us into the lunch break. So we are.
Now you remember when you go to lunch break to keep track of your AGRP neurons so that you don't do the same thing as the mice.
A good point. We'll see how we can do that. So I think, David, if you're in agreement, we would perhaps come back at 1:15, which would only put us about 5 minutes outside of our time, a reasonable half hour. We can take advantage of the fact that we're on Zoom. You can grab your lunch and come on and eat with your camera off, and we'll be able to move in at 1:15. And be able to take discussion. Thank you very much for the speakers this morning. Terrific talks, and we look forward to seeing everyone in about half an hour. David?
Yeah, thank you, Trevor. I think that works well for me. So a little less than 30-minute lunch break, returning at 1:15, and we'll jump right into Tom. All right, thank you.